Good afternoon. Those coffee lovers, please take your seat. Okay, good afternoon. My name is Raj Beshankiwar. I'm a scientist with the International Joint Commission, and I'll be chairing the session, Harmful and Nuisance Algae Blooms. Before I start my session, Dr. Whitman asked me to announce this. It's a BeachNet. Most probably all of you know about BeachNet. So if you are not have joined the BeachNet yet, so I'm going to circulate this notepad. Please write your name and your email so that we'll add you on the BeachNet. So, okay, let's start. I work with the International Joint Commission. My office is in Great Lakes Regional Office, which is in Windsor. It's a binational office. At the IGC, I focused on 3 or 4 areas. One is area of concern, groundwater contamination, beaches, and last but not least, harmful algal bloom, nutrient annex in the Great Lakes Water Quality Agreement. So this session, I think we have 5 presenters. Each one will have 12 to 15 minutes. I really appreciate you keep your talk 12 to 15 minutes so that we'll have time for questions. Before we start, I can safely say, because we all had our lunch, I think before the lunch, technically we talked most about the shit. So now we'll focus our shift from SHIT to harmful algal bloom. So very first speaker is, I know Eric, you told me not to say that, but Eric Wiegert? Perfect. So he's going to talk about New York State Department of Health's harmful algal bloom response protocol and tools.
So good afternoon, everyone. When I saw the preliminary agenda, I recognized that I was speaking after lunch during the time slot when your body's solvent is being drawn from the organ responsible for you to pay attention to absorb nutrients that you consumed. So I didn't know that it was after Callum, however. So please excuse me for not having nearly the quality of anecdotes nor humor. And certainly, I don't know if you were expecting me to have a New York accent, but Callum, so I met you in Green Bay, and I've been wanting to ask you to do this for a very long time. Could you just shout the word freedom? Okay. My name is Eric Wiegert. I'm with the New York State Department of Health Center for Environmental Health, Division of Environmental Protection, Bureau of Community Environmental Health and Food Protection, where we oversee a number of grant programs and regulatory programs, including the administration of the Beach Act grant in New York State. A bathing beach in New York State is a regulated entity, like a restaurant. It gets a permit, it gets inspected, and the regulations cover not just water quality, but also injury prevention and drowning prevention, which some of you may have heard me speak about last time that I was actually able to attend this meeting, which I'm very thankful to have my travel approvals done at the highest level. Always a great opportunity to be here in this organization, which I really, really respect and enjoy coming to. In part, I think because the way that I see the Great Lakes Beach Association is this melting pot, right? This multidisciplinary entity, people from all these different facets and specialties kind of come together to share ideas, to have some kind of a unified vision. And so the topic of my talk actually was a similar approach. So we saw what appeared to be an increase in cyanobacterial blooms, blue-green algae blooms, cyanohabs. What do you guys call them? Do they have a unified name yet? I don't know as if they do. And so in response to that, there was a multidisciplinary work group that was assembled in the department. And so we had toxicologists, and we had limnologists, and we had outreach experts, and then me, I would call myself a pragmatist, right? So trying to take from all those specialties and develop something that's actually useful for local health departments primarily, because those are the folks that are implementing our regulatory program. So how many people in the audience do we have here that are from a local health department, or say a Parks Department beach manager? All right. The rest of you, please bear with the presentation. It's not science, but there is some science basis in there. All right. So this increase in blue-green algae blooms that are occurring in New York State, it was at our inland beaches. We haven't had any of our beaches impacted by cyanohabs on the Great Lakes as of yet. We're very thankful for that. And just an overview of what I'm going to try to talk about here. Just a little bit of background on HABs, cyanohabs, and the health risks, what our New York State protocol is, and how we develop it, and how we implement it across the state. And so blue-green algae blooms, they occur when the conditions are right, right? So we're seeing potentially an increase in temperature, increase in nutrient loading, and it can have a tremendous impact economically and on recreational conditions. I think most of you here may be familiar with them. They can be quite unsightly, but even such, we've seen people actually recreating in blooms because they weren't aware that it was a health hazard. And so the public health exposure potential can be from ingestion, inhalation, or direct contact with the cyanobacterial cells themselves. So I think this is a critical point in shaping our guidance, and at least 6-7 years ago when we were formulating our guidance, it wasn't something that was commonly known. Most people were thinking that it was the toxins that the cyanobacteria blooms are producing is the primary health hazard, which it is still a health hazard, right? However, direct contact with the cyanobacteria cells themselves can still result in symptoms, be it through inhalation or primarily direct contact with the eyes or with the skin. So I'm going to read this really loud because it's bolded and there's an asterisk. Whether toxins are present or not, there is still a concern for health effects. And I already covered most of what's on this slide here. So dermal exposure pathway through ingestion, digestion, of course, incidental, inhalation, and the type of symptoms that you can get are like a rash, an allergic type of reaction, diarrhea, nausea or vomiting, and some respiratory difficulties from actually inhaling some of the cyanobacterial particles. So what have we been seeing in New York from a standpoint of blooms impacting bathing beaches? Well, we had some sporadic surveillance of these types of occurrences throughout the history of the department, but it wasn't until around 2010, 2011 where we started seeing an uptick. And I think for all of you scientists that are in the room, without me actually performing any type of statistical analysis, I think you could say that this is a statistically significant increase in the number of occurrences, right? Some water. So it crept up on us pretty fast. And so from going to virtually nothing to 74 beach closures in that short of a time frame, there was a need to develop some guidance and to roll it out, to get it out to the public health practitioners and beach managers who would need it. So there was a need for guidance, and we had this multidisciplinary approach in order to do it. So let's see. So while Callum was talking, I made this up. So now to prove a point, so I need to set the stage here. So let's just say there was a research institution on the west side of Lake Michigan, who are very well respected, but they're also known to have a lot of fun. And I think one of them has already left for the day, so I can take even further liberties here, whose laboratory might be actually named after me, the Environmental Research and Innovation Center. Where's Kim? Is she still here? She's not here either? Oh, so they named their lab after me. All right, and so, this can go really far. All right, so one night after being out at a Great Lakes Beach Association conference with everyone at an event where they're giving out free tickets for beverages, they decided to go back to their lab, right? And they had this great idea that they were going to genetically engineer for fun E. coli so that it would produce a pigment so that the E. coli could actually be visible. All right? All right. And then they had some kind of a silly lab accident, and they accidentally dumped some of it down the drain. It got introduced into the environment, right? And then, of course, it would spread, like, right, and then be passing genes and all that stuff that bacteria do, right? So then every time there was some kind of a sewage overflow or something, you could actually see the plume because all the E. coli would be expressing the pigment. All right, are you guys with me? Okay. It's a weird rabbit hole, but all right, here we go. All right, so now I need everybody here to close their eyes. Close your eyes. And I can see you all if you're not closing your eyes. Including you on the WeberNet. Hello. So close your eyes and take in a deep breath. Okay, and take in another deep breath. All right, it's just 3. I promise you it's just 3. All right, now I want you to all find your beach. You're on your beach, your favorite beach. You're doing what you do when you're on your beach. All right, I know what I'm doing. I'm sitting in my chair, I have a cold beverage, I'm watching my kids boogie board. I'm not at work. It's awesome, right? Okay, but then all of a sudden you notice the water starts to change color a little. Keep your eyes closed. Starts to change. Oh, wait, I didn't tell you. The pigment color, it's fluorescent pink. All right, so stay with me. The water starts to turn fluorescent pink. All right, and you know that there's a significant increase in the concentration of E. coli that's there, right? Right? And then you're at your beach and everybody's swimming and the water stays hot pink for hours, days. And then a public health professional comes and samples it. And then they're going to go analyze it. And then you're going to have the result. Does this make sense to anybody? I'm sorry, I was told once by a professor that visualization and sarcasm are a very useful learning tool. Okay? So what we've determined in New York State, right, is that you shouldn't be basing your public health prevention from harmful algal blooms or cyanobacterial harmful algal blooms based on toxin sampling. Because we know that you can have health effects without toxin. Right? And if you see it, chances are it's of a dense enough concentration that you could have health effects. That's a pretty safe bet. So why would you have public health prevention based on a toxin monitoring program? That's what I was hoping your response would be. So we do, however, have some analytics built into our approach, right? So you close a beach when you have visual evidence of a bloom. When the bloom is gone, you wait a day and then sample, right? And we're only analyzing for microcystins at that point. And it's almost serving as an indicator of all the other toxins, okay? And you have to wait a day because of the half-life and fate of other toxins that we're not measuring for. So there could be some residual toxins that are left behind. And that's not extremely well studied in some areas. So we've already established that you can get symptoms without toxins. We all know, right, that's been the theme of this organization for a very long time, that sampling and analysis takes time, right? And typically not all toxins are analyzed. So further coming into play, blooms are very dynamic, both spatially, temporally. They come and they go, right? But so does their toxin gene expression. So you might collect a sample for toxins, and it says that there are none, or that they're very, very low. And in a matter of hours, they could be extremely high, right? So, and then of course, it's not practical to monitor all waters at all the times. at all times. And so we've developed outreach materials so that bathing beach operators and park managers and local health departments and the general public have an understanding of what cyanobacterial blooms look like, and then they can put the ability to protect themselves in their own hands and close beaches before sampling, before consulting with cyanobacterial bloom experts. So we made a fact sheet for beach operators with some examples of what blooms look like, and to have an understanding of they need to close their beach to be proactive, and a number of other steps that are going to occur. And then for the public as well. So I'm not a millennial, I'm a little before that, but I'm also, what was I just saying this to, working in a section at the health department that used to have 11 people and now there's 4, and I don't know, I was a graduate student and a parent all at the same time, right? And then all of that other stuff. So I do like quick little soundbite pieces of information, right? So know it, avoid it, report it. It's pretty simple message. And then of course, mostly we're dealing with human public health in our agency, but blue-green algae effects aren't limited to people, right? It can be pretty severe in animals. Anybody had dog deaths in their states? Yeah. Yeah, and I think when I sat down in here this morning, there's a little table conversation occurring about pets, right? They're an important part of our lives, right? If you're in a local health department and you haven't had to contend with what happens, you know, when there's an animal death at one of your water bodies, it's a pretty severe response. So let's talk about. So we figured out that we needed to just have a basic avoidance message, right? So how do we deliver that to our local health departments? It seems like it should be pretty short. So what we did, we made a decision-making matrix, put it in a flow chart, right? But then, of course, as things evolved, it got more complex and started to look like this, right? So it is almost too complex to actually send out to people, right? So then the other alternative would be building a guidance document. It would have been pushing, I don't know, 30 pages with a bunch of series of skip patterns. I wouldn't want to send that out to anybody, right? Having worked in a local, actually, I would send it to EPA probably, I'm sorry. I wouldn't send it out to local health departments. So after we kind of finished the decision-making matrix and all these, and then realized that we needed to have supportive tools rolled in, right? So that we give the health departments the hot setup so that they can respond in the ways that they need to. I had to go home and update some computer software for my printer, right? So I was really stressed over how we're going to do this. So I'm updating the software, and it kept saying, well, if this, click here. If that, click here. I'm like, yes, yes. So I jumped in my car and I went back to work. No, I didn't do that. The tower was locked. But what we did was we ended up making this online interactive tool that's hosted on an intranet site that local health departments can access, right? And so the whole idea is that it's like you're browsing some kind of a wizard that can help you in your response, right? And that is linking to all the supportive tools that you need. So including identifying a bloom, what to do if you have a bloom in your beach, near your beach, or if it's somewhere likely to impact your beach, and then all these other resources that you need. So in order to help identify, to help sample, outreach templates, always extremely important, including examples for press releases where you just fill in the blank with the water body name and date, and you're ready to roll. And then the beach reopening and sampling procedures, which can get a little tricky at times. You know, when is a bloom really gone? And so we provided tools in order to have the local health departments be able to move forward with that. Other additional tools built in there are contacts, because a lot of it is all about communication. Again, beach operator information, and then how to report illness symptoms. So we have regulatory, epidemiological surveillance in New York State, where if someone claims that they got sick from a beach, there has to be an investigation. And that is done with a series of tools that we built and helped with a CSTE fellow who was with us the last couple of years, and then potentially reported nationally to CDC depending on the nature. And so I also recognize the risks of logging online and showing you guys these tools. So all I did was just grab a couple of screenshots. So it walks you through that same flow chart, but using embedded hyperlinks. And so based on the information that you have that you need to respond to, you follow the instructions and click, and it takes you where you need to be for your decision-making tool. And here's examples for public notification for beach closures and whatnot. As far as the deployment aspect, so we rolled it out. It's available on an internet site. And as you guys could tell from the number of beach closures that we've seen, we've had a dramatic increase in the usage of the guidance. And we believe that it's been successful because we haven't had any outbreaks associated with our regulated bathing beaches, despite a pretty drastic increase in the number of cyanohabs that have been impacting our beaches. So here's the key players who helped us build the tool here. Gina and Jim. Here's our interdisciplinary work group. We also get a lot of assistance from our New York State Department of Environmental Conservation, who have an extremely comprehensive surveillance program throughout the state, including citizen monitoring groups and lake associations. And then, of course, the local health departments and our regional staff, who are really the ones who are implementing the guidance, doing the hard work that it takes to protect public health day in and day out. And certainly we're very appreciative of them. And with that, I thank you.
We have time for one question. Time for one burning question. Yes.
You may have mentioned this. What was your criteria for closure?
For closure, if there's a visual signal, so if there's any visual indication of a bloom that's impacting the beach water.
Is that decision left up to the local beach manager, health department, or?
Yeah, so it's left up to the beach manager. We've provided them with tools so that they can recognize when there's what they believe is a cyanobacterial bloom that's occurring. So if it's even suspicious, not confirmed through photographic or microscopy or other lab analytics, the expectation is that they would close the beach, especially if it's on a water body that's prone to blooms. Yeah, and so we've provided tools so the beach operators and their lifeguards can recognize blooms.
Sorry, we'll have after the end of the session, please.
Sure, I'll be around. Thank you.
Sure, yeah. The next one is regarding genomic analysis and detection of microbial communities. Casey Kelly, right?
That would be Kasia.
Kasia, sorry. Please welcome.
Is the cookie thing good? Okay. The microphone? Okay, I think I'm good. Richard, thank you for your note. Before my presentation, Richard said if I say the joke at the beginning, I'm not going to be that nervous, but I couldn't come up with the joke because I'm so nervous. So there you are. I can just say, hey, maybe hi to my mom instead. Cześć mamo, mam nadzieję, że patrzysz i że trzymasz kciuki. Okay. I work for United States Geological Survey, a Great Lakes Science Center, and today I am going to be talking about Cladophora. And first of all, I would like to connect with the presentation that we heard earlier this morning. It's the one that Clarissa, if I pronounce her name correctly, delivered. And she so nicely showed us how beneficial and how grand beneficials and advantages we can get from DNA-based analysis if we can successfully apply them to any kind of trouble that we have in our beach ecosystem. She was talking in particular about the connection with water and the fecal matter and using the DNA. I am going to be talking about the DNA from Cladophora, and Cladophora is a very complex ecosystem, and that's why this all gets just quite complicated. So again, back to Cladophora. As you can see here on the picture, I'm sure that most of you is very familiar with Cladophora. You see it all the time at beaches. It grows very well on any hard surfaces. It attaches well to the rocks. It grows on the break walls. It's everywhere, underneath the water surface. And during summer months, it frequently sloughs off and ends up on our beaches, where it's cause of all kinds of different troubles at the beaches. Now, there is a lot of people who do a lot of work on Cladophora. scientists in the office where I work, we've been working with Cladophora for like probably 15 years. And there is Mike Sadowski laboratory and Greg Kleinheinz. They are all working in so super complicated directions to solve the Cladophora problem. Okay, to start with, so I wanted to maybe change the theme. I know that we are in harmful algal blooms theme and everybody is talking about bad stuff, but there is something that we can call a Cladophora conundrum, if I say it in English. It's probably conundrum, okay. Anyways, lots of people that are mostly familiar with the negative effects of Cladophora, with the fact that this is one of the 10 very important environmental stressors that affect the Great Lakes. There is the potential of bird botulism, and it usually has very high concentrations of fecal indicator bacteria, and it can contain pathogens. It's also a threat to the invasive species, and like we have here on the photo, a little piping plover, and somebody also had the presentation this morning about the endangered piping plover. So we see the connection here that obviously it affects so many different directions. And in Lake Ontario, it does already cover, up to 40% of the benthic areas. Now, on the flip side of Cladophora, and this has been kind of the topic of the pretty recent research, and maybe people starting to look from different directions, not only from this very negative standpoint. Cladophora is a native alga for Great Lakes. It is not invasive species. It did not get here by chance. It's always been here. It is the environmental engineer, and it has the ability to thrive in this, freshwater ecosystem. It can grow in a marine ecosystem. It's a super capable alga that is found here in the Great Lakes. And what we can see is from little bits of preliminary, usually metagenomic studies, is that it does provide a good nursery for the juvenile fish. It also is a very good part of the productivity primary production in the Great Lakes. And it also has a very vast community of bacteria and epiphytes. So going back to the negative side, something that we all are very familiar, just on this slide here, I have just a few examples of the studies that were conducted on decaying Cladophora mats that were deposited and accumulated along the shoreline. And as we can see here, this is all published work of the colleagues. Very, very high concentrations, not only for fecal indicators, but also for pathogens can be found in Cladophora. And this is something that we are so very familiar with. Now, here, as the objective of our research to go into the Cladophora genomic studies was to focus not only on the negative, but also add up this positive effects of Cladophora, just to increase our understanding what these biological communities in Cladophora, what is their function, what are they doing there, and what is their composition. And perhaps with that type of understanding, we could have the better grasp what's its role in the ecosystem, and also come up with maybe perhaps some solutions for the beach managers. So here in particular, the first step was to develop and optimize a DNA-based method. So because there are not really many consistent methods for very complex substrates such as Cladophora, the first step had to be to go into the sample processing and optimizing DNA extraction protocol, so then we can use it most efficiently for metagenomic research. Secondly, with this extracted DNA, we were able to validate, pretty much our protocol, our sample processing and extraction protocol by using a qPCR assay, and this one was targeting Bacteroidetes. It's a group of bacteria that just is so dominant in the aquatic and terrestrial habitats, so it seemed very appropriate that we would be able just to test this qPCR assay on our DNA, just to see if the DNA is applicable for the molecular work. And thirdly, the whole idea is just to identify all the biological communities, all the assemblages that are associated with Cladophora. And the technique used here is the shotgun sequencing technique. It's the whole metagenome study, which encompasses pretty much all and every organism that is associated with the Cladophora. It's not only bacteria, but also the higher organisms as well. So if you would find the sequences there that belong to macroinvertebrates, fish, humans, that method is that is the method. So in 2015, we were collecting samples from very different environments because few of those were associated with the urban ecosystems. These were beaches in very busy areas, urban areas. So there is Calumet Beach in Chicago, Racine, North Beach in Racine, and then George Park in East Chicago. But also there were two locations. One of them is Sleeping Bear Dunes, and the second location, Portage Lakefront. This is very close to our office. And these are these belong to national parks. So there was kind of variability here with choosing the sampling locations. We ended up collecting 36 algal samples, and we focused on growing, living, attached algae. So we were pretty much pulling the filaments off of the rocks and the breakwalls, and we were not taking any samples of the algae that already got detached and was like accumulating on the shoreline. We were not going into this. We were just focusing on the living Cladophora that was still attached. And here I would like to point out, because every time we would bring the samples to the laboratory, we were proceeding with this visual inspection under the microscope, and the samples were so variable, and sometimes you would like lose your faith that this is still Cladophora. But then looking under the microscope, it looks like some of the filaments were just so strongly encrusted with diatoms, and there were so many different assemblages of macroinvertebrates and meiofauna that it just really changed the appearance of the algae itself. Now, algae from Sleeping Bear Dunes was especially different because these were collected by the scuba diver, and she had to. It's very hard to pull Cladophora off of the zebra mussel beds in Sleeping Bear Dunes because there's no rocks or breakwalls. Usually, all these Cladophora mats are just growing attached to the zebra mussels beds, and they are the attachment is just so strong that for her to collect these samples, she pretty much had to pull like half of the zebra mussel bed along with the sample. But that's just for our, visual, amusement. And in our methods development, and again, I wanted to point out, because there is no consistent processing technique for Cladophora, as it is probably for a lot of very complex substrates like soils and sands. We were focusing here on, I'd say, 4 elements to test, and try to optimize it for obtaining very good quality DNA for the further analysis. So first of all, we were varying the weight of the algae, so we were trying to take, and this is going to become much more clear when I come to the next slides when I explain the technique, but so just bear with me here for a moment. So we were trying to choose different amounts of algae. Then after we were processing the algae, we were testing different amounts of the pellet that we were using for DNA extraction. We were using this commercial kit from Mobio. It's called Mobio Power Biofilm. And maximum amount that you can input into your tube is 0.2 gram, but it's not always the optimal amount for your DNA extraction. So this is one of the elements that we also varied in our search for the perfect technique. And then also what we were doing with such a big effort is using pellets from rinsed and unrinsed algae. And this is going to become more clear in a moment. So rinsed and unrinsed algae. And lastly, we were applying very particular modifications to the manufacturer's DNA extraction protocol just to increase our efficiency and also get rid of the amount of inhibition that we might have had in a way. Okay, there's my question mark. So this was all to be tested for optimizing our processing technique. And this is the workflow, how it all looked when we were bringing samples to the laboratory. So at first they were all like kind of draining on the net just to remove all the excess liquid. From here we would take a sample of unrinsed algae, okay? So there would be unrinsed, unprocessed, didn't do anything really with this. But then a portion of the algae would go through triplicate rinsing. So we would just take, like there was 30 grams of algae that we would like swirl in the water. This was pre-BS actually. But then transfer from one of these cups to another. And as you can see, the color of this water, and also probably you don't see it because if this was dark here, you'd see it better. But there are like lots of white points, especially in the first photo. And these are very small amphipods that were just swimming, around in the Cladophora, and they were so common from like one of the locations. And then the photo from the bottom, it was one of the locations that always experienced so much diatoms attached to the algae. So whenever we were trying to rinse it, the rinse water was coming out so dark every time. Now the whole idea about rinsing was that there is the community with Cladophora that is very tightly bound to the filaments, and probably somehow, I'd say magically, it's not scientifically said, but it's very tightly bound to the filaments, and you cannot get rid of it very easily. But then there is another community that is very loosely associated and just kind of lives off of Cladophora ecosystem. And this community you can rinse off very easily. And I'm sure that like for beach managers, always the question would be like if you worry about your E. coli or pathogens or any of the bad stuff, well, you would probably be always asking, is it tightly bound or is it with this loosely associated material? Can it wash off very easily and get to my water and affect my beach? Or it's okay because it just grows with this algae and no one really interacts with algae that tightly. So we thought that this aspect was very important, try to distinguish rinsed and unrinsed communities. So now coming from here, so after rinsing process, the next step was elutriation. That this is how we say it in our world, elutriation, and it's nothing less than just taking a sample of algae, mixing this with the PBS phosphate buffer solution, okay, or saline, and then you shake it very vigorously for 2.5 minutes, and you do it 2 times. And your next step is we were using these very useful filtration bags. So the whole idea is that when you are shaking this, the theory is that all the organisms that are present in Cladophora, they would be removing themselves to the liquid. So then you're ending up with all that you need, right, with all your cells and DNA inside of the liquid, and then Cladophora just stays behind. So the filtration bag helps you kind of squeeze out all these juices out of Cladophora with all your organisms and DNA, everything that you need for your further processing. So after that, we were taking the supernatant and this liquid that would theoretically contain all the cells, all the organisms, and all the biological matter, and we would spin it down in a centrifuge, and we were obtaining the pellets that were very different for different samples and from different geographical locations and also over time. So that was kind of like interesting to see that sometimes we were getting really super fat pellets and sometimes they were just very small. And of course we were just processing rinsed and unrinsed algae at the same time. We were varying the amount of algae that we were using. We knew that we had to use less than 0.2 gram for the DNA extraction. So now again, just a reminder of these 4 elements that here we were testing for optimizing the technique. So as the result, we came up with that we pretty much have to use the minimum of 25 grams of algae, that our optimal pellet is 0.15 gram, and the good DNA yield and purity we're getting from unrinsed algae. And also we had to go with the modifications to the, this is very technical, so I just don't want to be explaining this too much more, but there were 3 of the steps that we optimized in the manufacturer's protocol just to get better results on DNA. So these are the results from 36 Cladophora samples, and as you see, there are pretty good DNA concentrations, and 260s and over 280 ratio pretty much indicate that there was good, there was really probably not much of the inhibition potentially, and there was lots of DNA always in Cladophora collected from Portage Lakefront, despite the fact that pellets were always very small from over there. So that's just a few of the highlights from this. Now, as the validation, whether or not our DNA is appropriate for the molecular test, we use this qPCR assay for Bacteroidetes with GenBac3. And as expected, we did detect bacteroids in all the samples. There was lots of variation, and two of the locations that are geographically very close to one another, they appear to be correlated. Otherwise, we did not really experience any problems with this DNA obtained with our optimized method. This here, and again, just to remind you, when Clarissa said that she had 80 million reads, I could really relate to this because we had lots of reads, as you see. So the amount of reads that you get from shotgun metagenomics technique are very high. So, our lowest is like 26 million, and then our highest is 16 million reads. And this just gives you the overview of how many different organisms are potentially associated with this ecosystem. It's just so super overwhelming amount. And in the little table on the right, there is the reference to several of the substrates that I was able to look up, like, for example, in soybean rhizosphere or river plankton or in Microcystis bloom, when people were doing the same technique, shotgun sequencing, they would be getting probably about 10 times less reads per sample. So we are thinking that this Cladophora ecosystem is like really rich in biological communities. The last element here is the Cladophora bacterial epiphytes. This is one of the recent studies from Zulkifly and Graham. They really are working on the Cladophora, but this only concerns the bacterial communities and nothing else really. So moving on the right, you have pretty pictures from our microscopes. So visually speaking, if you looked at Cladophora, you would always say, wow, there's so much stuff here. And you would always see so many macroinvertebrates and protozoans and diatoms and just such a rich, diverse community. Now, when you go the DNA-based method, you discover that like super vast majority of the organisms that are associated with Cladophora is actually bacteria, and rightfully so, that provides lots of surface area, Cladophora provides lots of nutrients, so bacteria are just thriving in this ecosystem. So on the other hand, like any type of cellular organisms, that's pretty much like about 10% of the whole metagenomes study. And just lastly, as the conclusion, I just wanted to say that this DNA extraction protocol was successful for these genomic applications that we tested, and we discovered very, very vast community of organisms that was associated with Cladophora, which was a very encouraging result, something very exciting as well. And since now we kind of have the idea of who's there, meaning what kind of organisms are inhabiting this ecosystem, for the future analysis, we would like to focus on the functional characteristics, meaning what are they doing there and what is the role in the ecosystem, and also how the composition of these communities and their function can help us solve this Cladophora conundrum. So that would be it, and I thank Emily for scuba diving for the samples and Stefan for shipping them, and especially I thank Ashley for the English word conundrum. Thank you.
Thank you, Professor. Thank you. I don't think we have time for a question, so I'll introduce the next speaker. I'm excited. First note is I try to keep this session more sanitized, free of bacteria and fecal coliform, but they're following me anyway. So And I'm very excited about next speaker, Ravina. She's from Canada, my neighbor from London, right? University of Western Ontario. And she's going to talk about nutrient transport from the septic. So again, septic there.
Unless you want to give Mark's talk. It was really cool. There it is. Perfect. Cookie? That's right. And then, yep.
Good afternoon, everyone. My name is Sabina, and I'm from Western University, and I'm going to share some findings from my research and talk about nutrients transferred from septic systems. So we all know that nutrients pollution is very great threat to ecosystems of the Great Lakes. And sources of nutrients, some of the sources of nutrients are displayed here, but today I'm going to be talking about the nutrients derived from septic systems. So why septic systems? There are hundreds of thousands of septic systems installed along the shorelines of the Great Lakes, and they're a pretty common way of the disposal of human waste. And 25,000 new or replacement septic systems are being installed annually in Ontario. And it's been reported that 20% of septic systems estimated to fail due to poor design or poor maintenance. So from the design of septic systems, they are supposed to deliver nutrients to the subsurface. So they're supposed to leach through the groundwater and essentially infiltrate to the groundwater. And there was a lot of research done on the transport of nutrients to the groundwater. However, most of the research that have been done, they were just focused on their immediate proximity from the septic tile bed. So we still don't know whether the nutrients are delivered to the Great Lakes and whether it's the nutrients derived from the septic system is a threat to the lake water quality. So that's what I'm trying to understand with my research. From previous studies, it has been found that phosphorus can be very mobile in the calcareous soils, such as sand. So that makes beach aquifer very vulnerable for potential phosphorus mobility and potential phosphorus discharge to the lake. So with my research, I'm trying to answer the question, will septic-derived nutrients be discharged to the lake, or if not, why wouldn't they? So let's look at this picture of the beach. This is a pretty dynamic and complex environment. We have an exchange of water between groundwater and lake surface water. And due to this mixing of groundwater and surface water, either due to the seasonal water level changes or due to the wave activity, we have a so-called reaction zone, because due to the mixing of the water, it drives the redox and pH gradients, which can affect the geochemistry of pollutants, including nutrients. So I'm trying to understand how this reaction zone affects the nutrient plume derived from the septic system before it gets discharged to the lake. Previous studies that have been done on this reaction zone in marine settings and riverine settings have been found that that reaction zone plays important role in the transformation of pollutants in those environments. So with our research, we're trying to see how the role of the reaction zone in the freshwater environments such as Great Lakes. So my main research question and objective is to what extent septic system nutrients derived from septic systems being transformed before they discharge. And I'm trying to understand essentially the movement of nutrients along their pathway and the transformations that they might undergo in the reaction zone. And this knowledge is particularly important because now although there were a lot of reports on the state of the Great Lakes that acknowledge that septic systems might be contributed to the nutrient pollution in the Great Lakes, there is no quantitative information about the loading of nutrients from the septic systems to the lakes, and therefore this knowledge is needed to develop the management and regulations for the septic systems control along the shorelines. So in order to answer the research question and kind of get an insight of the nutrient behavior in the subsurface, we picked our site on Lake Huron. That site has a public washroom. It's a homogeneous sandy aquifer. And we installed the cross-shore and long-shore monitoring transects and did the monitoring of the nutrient plume in 2014 and 2015. So as for our monitoring, so we installed the cross-shore transect. That's the picture of the cross-shore transect. And then we installed the piezometers, and we collected our groundwater samples using the multi-level samplers as well as the drive points. The drive points were used at the dunes to get to the deeper depth. And we also collected the sediment samples to understand the phosphorus absorption behavior. So during our preliminary studies, we tested 3 different sites along the Lake Huron, and we noticed that the site that was adjacent to the active public washroom, the concentrations, the groundwater concentrations of nitrate on that site was much higher than other sites and also higher than surface water concentrations. We also performed a along-shore study of the plume to determine their along-shore variability, and we found that the nutrient plume is very narrow, and that allowed us to focus just on the one cross-shore transect, and now let's look what's happening in that transect. So first, phosphate plume. So here you can see the conceptual diagram of the site, where on the x-axis you can see the distance from the septic system, and then the y-axis is the elevation. The black line represents the sand profile, and then the blue line represents the water levels. So the groundwater flows toward the lake, and then the concentrations of the phosphate observed in August 2015 were as follows, and the scale is a log scale. So we found that the groundwater concentration of phosphate was more than 400 micrograms per liter, and that's way higher than surface water and way higher than the current Canadian water quality guidelines for the protection of aquatic life. So further, to study the mobility of the plume, we looked into the groundwater velocity, which is 230 meters per year, as well as the time when the septic system was built, which was approximately like 40 years ago, and then as well as the current location of the plume and the current concentration. And we found that the phosphate plume is moving slowly towards the lake, and then based on the parameters that I mentioned above, we've calculated that it will take 13 years for the plume to reach the lake. So, then the important question here is what makes the phosphate to move slower, and it's due to sediment interactions between phosphate and different minerals that are present in the sediment phase. And the 2 main processes that would control the slowing down of the phosphate transport would be sorption and precipitation. And it's important to distinguish between those 2 processes because sorption is a reversible process, meaning the immobilization of phosphate is more like temporary, versus precipitation on particular on the secondary minerals would mean that phosphate might be immobilized more permanently. So in order to study those 2 processes and phosphate interactions with the sediments, we collected some sediments, and then from preliminary analysis, we can see that phosphate correlates well with iron and aluminum that is present in the sediments. And according to the literature, phosphate indeed reacts with iron and aluminum and to precipitate as phosphate minerals. So first step would be to perform the sequential extraction analysis to identify between labile and non-labile phosphate forms to distinguish between precipitation and sorption reactions that would control the phosphate mobility. So now if we switch our focus on and look at the nitrate plume, we can see same conceptual diagram, the groundwater flows to the lake, and then we observe the groundwater concentrations of nitrate more than 100 milligrams per liter. And again, those concentrations are way higher than surface water and the Canadian regulations. It's also important to see here that nitrate plume moves faster than phosphate and reach the shoreline of the lake. However, the concentrations get decreased next to the shoreline, and which again implies that the reaction zone might be occurring. And it's important to understand whether this decrease in nitrogen concentration occur due to the dilution or is it a reactive transformation. So now if we look at the two diagrams of the nitrate and chloride profiles near the septic system, we can see that chloride is being the conservative tracer. So you can see if the profile of chloride and nitrate match well, we can tell that nitrate behaves as a more conservative constituent. So we can see that near the septic system, the profiles did match pretty well, and then the correlation is, they have a good positive correlation, so that tells us that near the septic system, nitrate behaves as a more conservatively. However, if we look at the same profiles near the shoreline, we can see that the profiles don't match here, and the correlation is poor compared to the previous one. And that again indicates that there might be some transformations of nitrate that might be occurring in the reactions that I was telling about. So our previous student has been doing the research on the wave-induced groundwater flows at the same beach, and he found that the groundwater flows in that particular beach is pretty dynamic, and there is a lot of recirculations going on, and this recirculation caused redox and pH gradients to occur. So this might have an important impact on the transfer of nitrate, and that might cause the decrease of nitrate concentrations. So the next step with that would be to determine whether it's dilution or reaction such as denitrification causing the decrease of the nitrate. And in order to do that, we would do the stable isotope analysis to determine the denitrification, as well as combine the physical and geochemical modeling together to better understand the behavior of nitrate in the subsurface. So the last question was that remaining for us, it was whether those nutrients that we observed there are indeed coming from the septic systems. So in order to do that, we did the artificial sweetener data analysis with the help of our collaborator, Dr. Tasek from University of Waterloo, and we analyzed the 4 common artificial sweeteners, acesulfame, sucralose, cyclamate, and saccharine, and we found that 2 of them, acesulfame and sucralose, behave more conservatively, versus the other 2 are being not detected. And in the literature, the previous literature has been seeing the same results, where cyclamate and saccharine are considered to be more subject to absorption and biodegradation, and therefore they're less effective. However, if we compare the profiles of the 2 artificial sweeteners with the profiles of nitrate and phosphate, we can see that artificial sweeteners can be a good sort of second confirmation that the septic system, the nutrients that we see there are coming from the septic systems. So the conclusions of this work is that we see the elevated concentration of nitrate and phosphate coming from the septic systems and reaching further down gradients of the septic tile beds. The artificial sweeteners indicate that the nutrients that we see at the site are derived from the septic systems. The phosphate migrating slowly towards the lake, however, either precipitation or sorption reactions might slow down its transport. Nitrate is transforming next to the shoreline, and the groundwater flow dynamics can be an important contributor to those transformations that we see, particularly in the next to the shoreline. So I mentioned the work that I'm going to do in the future, and I'm happy to share it later next year maybe with you guys. So thank you for your attention, and thank you for our collaborators and sponsors.
We have time for 2 questions.
Could you tell us what the nature of the soil was? Was it sandy? Was it clay? Was it anything about how rapidly the soil would transmit?
Yeah. So the question was about the nature of the soil of the site. Yeah. So the site is mainly sandy. So it's mainly of the, as I mentioned, it's homogeneous sandy aquifer. And that's the reason we chose that site. First, because it's easy to install our equipment. But second, most importantly, because previous studies have been found that the phosphate is being more mobile in those conditions, in the sandy aquifer and near-neutral pH environment. So we wanted to test that.
One last question. Yes, Eric?
Any thoughts on differentiating between organic or inorganic phosphate?
The question was about the differentiation between organic and inorganic phosphate. No, we haven't looked into that yet. I guess we're more interested in inorganic phosphate as of right now. Yeah.
Thank you, Sabina. Give her up. So, Shannon, I thought we had enough of qPCR for last 2 days. But apparently there is a very high public demand here. So, Mark? Yes. 15 minutes.
Good afternoon, everybody. Mark Citriglia from the Northeast Ohio Regional Sewer District. We are the largest wastewater utility in the state of Ohio. We are the largest point source, as everybody knows. So one of the things the Regional Sewer District wants to do is we want to be part of the solution, not part of the problem, and everybody points to us. So one of the things that our laboratory has done is really got involved with the Great Lakes Beach Association, which is a great association. And we've started doing a lot of work with qPCR, beach monitoring. Actually, we've got into harmful algal blooms. We've also worked with a few colleges here in Michigan, from University of Oakland with David's log, Judy Westrick from Wayne State University, done some work with Lake Superior State University. So it's been really great networking at these conferences and making connections and doing some work. So what I'm going to do is I'm going to talk a little bit about how our laboratory has decided to use this qPCR tool for cyanobacteria. What I'm not going to do is talk anything about qPCR because we had a full day of it yesterday. But I will talk a little bit about what we decided to do. I'm going to talk a little bit about the methods for toxin analysis that are out there real briefly, talk about our workflow in our laboratory, how we differentiate samples and how they should be analyzed. I'm going to talk a little bit about the CyanoDetect assay, which is made by Phytoxygene or Diagnostic Technologies. Greg Ford is here somewhere as a representative. And then I'm going to talk about the data that we've generated ourselves and how we're using it, along with some comparison data with another laboratory. And some of the issues that were brought up in the qPCR section on the E. coli assay, we've kind of seen some similar things in our method validation with extraction techniques and just problems with qPCR in general. So basically right now, the majority of the methods being used for cyanotoxin analysis is really the ELISA method. And there's 4 different toxins that can be analyzed by ELISA, cylindrospermopsin, microcystin, saxitoxin, anatoxin A. Ohio EPA last year, because of the big bloom in Toledo, came out with their own method. Just this year in August 2016, US EPA published EPA 546, which is very similar to the Ohio EPA method, has a little bit more quality control in it, very much similar to the quality control in the LCMS method. Happy to say that our laboratory was involved in the study. We did a lot of work with it. The next method is the LCMF method, which is much more sensitive than the ELISA. It also gives us the different conjugars. Right now the method is set up for 6. We have the ability at our laboratory to analyze for 14. Very expensive test. I mean, you're talking about $500,000, $300,000 instrument to do it. So sample analysis running from $300 to $500. ELISA, in Ohio, the price for ELISA has kind of come down because of the Ohio EPA has mandated that everybody's doing ELISA. Much cheaper to do more analysis. So some of the problems with harmful algal blooms right now is a lot of the cyanobacteria can produce multiple toxins. Just by identifying it alone doesn't tell you if it's toxin producing or what toxin it is going to produce. There's also some skill involved with the bacteria identification and enumeration, and there's time involved in it. So we have been, part of our procedure, our workflow right now is when a sample is collected, we'll do some sort of algae identification. And depending on the sample, it could be some difficulties in identifying it. So if it's a very dense sample, it's pretty quick. We can take it to our centrifuge, pellet the sample, take a little drop out of it, put it under the microscope, then start enumerating and counting. If it's not a very dense sample, well, it takes a little more time to concentrate. I mean, we can stick it in a 1 liter jar, let it concentrate, and then pull off the supernate, so it could take 2 to 6 hours to even get something where we can get enough bacteria in there to really kind of enumerate. Once we've kind of identified the bacteria, we say, hey, it's a cyanobacteria, then we would call our client and say, would you like toxin analysis, or just knowing that it's present there, is that good enough for you to make a decision? So if they chose for us to do a toxin analysis, we'd already start this freeze-thaw process just in case. Go ahead and do the analysis. And most clients, we tell them, hey, there's 4 toxins, which one do you want us to do? And everybody says, well, microcystins, so we always do microcystins. So that's our current workflow. One of the things we started to do in 2012, we started doing the ELISA. We started to support some of the local health departments. And so our need to do harmful algal blooms, not only Lake Erie, but some of the inland lakes had increased. So our workflow, we were saying how we're going to get through these samples. So our biggest thing was to look for a way that we can process samples a little bit quicker. So we tried some stuff with our enumeration, like using membrane filtration, filtering down a sample, doing the identification. Some of the problems with that is if you got too much vacuum, you break the cells. Cells are on top of each other. If there's a lot of turbidity and stuff in there, that makes it difficult. We tried some tangential flow filtration, which is pretty good. You can concentrate a sample relatively quickly. You tried some vacuum-assisted utermal chambers to help with it. So we tried all these different things. Still, it was time-consuming. We knew of qPCR, and there was research being done on qPCR from SQURP, USGS, Water Research Foundation. We're all doing work on qPCR. We happened to be at a conference in Akron, and Mark Van Esten from Diagnostics Technology was talking about his qPCR assay. And it sounded pretty interesting. We decided to give it a try. So some of the different things that we know, and this is really no different than kind of on the bacteria side, with the qPCR, we know that the cyanobacteria is there. When we do algae identification, we can confirm it by morphology. Another weird thing is there's really no direct correlation between the number of genes found and the number of cells. There's not a direct correlation there. The good thing about qPCR, we can know if the toxin producing gene is there, so you could have, the toxin may not be produced, but the potential is there. So understanding that is pretty good. Since we've had experience with qPCR, it wasn't a big deal for us to to utilize it. One of the bigger things that we had was just the time that it took to do the algae ID. So what we did in 2014 is we kind of started experimenting with it. 2015 we did some more samples, and now we have pretty much a new workflow in 2016, and I'll show you that in a minute. So what is the cyanotex assay? It's a multiplex qPCR. We can look for total cyano. We can also has an internal amplification control, which good quality control parameter. And then we can look for 3 toxin genes. It comes in 2 kits, comes with standards. It was kind of made to be, come out of the box, take it, run it, and analyze it. We've tested it on 3 platforms. We have a Second Smart Cycler, we have a Step 1 Plus, we have an ABI 500. It runs just fine on all 3 platforms. There is a slight adjustment that you have to make on the Step 1 Plus because of some of the filters, but other than that, it was pretty easy. So this is what we ended up with our workflow. Samples will come in, pretty much take that sample right away and we would filter it, similar to your crude extraction method for E. coli. Filter it, take it, put it in our bead beater, disrupt those cells, take the top off of it, stick it in our thermocycler, amplify it. And the great thing about it is we, not only know that because we can do total cyano on all 3 of the genes at the same time, we can tell our client, yes, you have total cyano there, and we found gene presence, and right now it looks like we see the saxitoxin gene, not microcystins. Would you like us to look for saxitoxin instead of microcystins? So it's kind of helped us identify stuff and being able to give our clients a little bit better idea of where to go with their analysis. So this is our current workflow right now, and pretty much every sample that comes in for toxin analysis, unless they really want to know. And there's a few people that still want to know what the predominant species is, and we'll still do the identification. But most of them want to know, is it total cyano and what possible toxin? So there's a couple different ways you can run the method. You can do a direct sample analysis. You know, just take some, stick it into a BioGX bead tube, bead it for a few minutes, centrifuge it, pull off the top and run it. Filtration, that's what we like. It's very simple, very similar to what's being done with the E. coli and enterococci method. You can also do centrifugation. We tried that. It works okay, but really we've stuck with filtration. And I'll show you some data on some of the stuff that we've done. Another reason filtration is easy, there's so many different ways to filter. And the great thing is that we can send syringe filters out with our field staff, and they can just filter it out in the field and bring us the filters back, which is great. So this is just a little bit about filtration. We use a 0.8 micron, where for bacteria we're using 0.45. We tried to go down 0.45, but the algae is a little bit bigger than that. It clogs the filter too quick. So 0.8 seems to work really well. So here's a quick study that we did doing the direct analysis, where we just took some sample, we beaded it, and then we analyzed it versus filtering some sample. And you can see, except for one occasion, the filtration gave us higher recovery and yield. Both on we have an internal culture of microcystins. We also have an anabina culture. And so those are kind of our control, and we would do stuff like do a cell count and then try to do a gene count on them. We also decided to do purification because there's still a lot of talk out there that says, hey, you really need to purify this. And so we did a study where we tried our crude extraction versus the purification method using a MoBio kit. Of course, the purification increased time, increased cost, and really what we wanted was a quick, rapid method. But we decided to let's take a look and see, do we gain or lose any. So we ran a series of samples. You can see that they kind of correlate really well. They go up and down, except for a point. The correlation really isn't too bad, but the big difference is the slope. The slope of the samples that went through the full extraction, cleanup, or whatever, much lower. So really, if you're going to try to make a decision, it's always, quick way to do it, crude extraction, make your decision on the beach, there you go. Calibration, so a couple other things that we wanted to do when we got this method, being a laboratory that's accredited, we like to have a lot of quality control, a lot of checks, and this thing was packaged that would give you all these standards, and you just take these standards, calibrate it, run it, and recheck yourself against the same standards that you calibrated against. Well, in an environmental lab, we always like to check with something that we didn't calibrate against, so we just chose to take their top standard, do serial dilutions down it, generate our calibration curve, and then use the standards that came with the kit as a readback. So it's not, it's not like we have a full independent source, but it's better than just reading back your standards. And so we did calibrations with their standards, and we did calibrations doing serial dilutions. And as you can see, regardless of which way we did it, they were really very good. They correlated very well, almost looking at over here in the corner there. Their slopes are almost on top of each other, very tight. So, we had a lot of confidence that the standards that we were getting were good and reliable. It would be nice to have an independent source somewhere that was quantifiable to use. Method quality control, this is what kind of comes with the standard. It's got, you know, we have to get a 995, you know, we follow the same QPCR stuff with our slope, you know, 90 to 100%. Good thing it does have an internal amplification control, and this has been helpful for inhibition. They gave us a mixed DNA standards. They're just started offering now a mixed independent standard that we've been working with them testing. So method validation. So there's another laboratory out there that we're working with. And so we decided, hey, let's do a validation study here with them. And some of the things we did is let's compare our calibrations and let's compare what the vendor came back with. The vendor came back and says, hey, you know, if you run our high standards, you could about get 18 CTs. This is their standard deviation. Well, we did about 15 calibration curves. And this is the data between two labs that we kind of put together. It really wasn't bad. We correlated very well. You know, about 1% RSD is kind of where we were going with each of the different standards. So between us and our secondary lab, things look really good when we just were running standards back. So that was phase one. The other thing we did in phase one is we took both the slope and the intercept of all our calibrations that we did, kind of compiled them together, and we wanted to see, are they significantly different or not? And so the first two, the first lab one, you know, we did one calibration, didn't have any problems. And the second lab, the second calibration, things were really good with the first two calibrations. Everything was, you know, was not significantly different. They had an instrument problem. They ended up recalibrating all their dies, recalibrated that instrument again, and things really changed. So you can see, like on the total cyano, you know, we were getting a slope of about 3.5, and all of a sudden they were getting a slope of 3.2. And this kind of will come apparent as we started analyzing samples, that then there was a significant difference between the samples that were analyzed between each curves. So they did have some sort of problem with their calibration on the second set. So that really tells you the importance of having an independent check, something that we can check back against. But just two labs, you know, just looking at slopes and intercepts might not have picked that up. So here's some interesting stuff. After we did all this calibration work, we decided to go get some samples. So Grand Lake St. Mary's, everybody knows where that is, from Salina. Then we got another one from Cadiz, and then we had some of our local beach samples. And so two laboratories, we each took 10 samples, both of us extracted them, and then we analyzed them to see what was going on. Basically, you can see that there was a huge difference from laboratory one from the stuff from Grand Lake St. Mary's. First lab was much higher, and then the Cadiz samples, we were much higher. And then the other one that was processed by us, that we sampled and processed ourselves, everything looked really good. So this was kind of really confusing to us, but we know how heavy that sample is from Grand Lake St. Mary's, so it didn't surprise us too much. So then again, we went back and we did a phase 3 study where we got the same samples, and this time we sampled everything. We put it in a churn, we then aliquoted out 20 samples. Everything looks pretty good. Samples correlated. We extracted them, sent them to two labs. Everything looks nice. So we know that, you know, extracting and processing is a very important part of this assay. But if it's done consistently, we get pretty consistent results.
Mark, sorry.
Oh.
One slide, please.
One slide? Okay, let me go to this slide here. This one I think is the most important one here. This here is we're tracking for a local wastewater treatment plant. We are tracking their total cyano cells, and we're tracking their microcystin genes and the saxitoxin genes. This plant has never had a problem with saxitoxin. We started seeing throughout the year, getting close to October, all of a sudden the total cyano started rising, and also the saxitoxin genes started rising. And we started getting hits for saxitoxin just after that. So this plant here, who was normally doing microcystins on a regular basis, really wasn't aware that in their raw influent they were getting hit by saxitoxin. Now their treatment technology is working well where they haven't had it in their influent, but now this last sample that we pulled in early October, they start to get a spike in microcystins. So at the very end, so we're kind of seeing, and we'll see as we continue through October, do we see the saxitoxin community going down, that gene, and then the microcystins popping up. So I think as a screening tool to kind of understand what's going on in your water system, I think it is worthwhile in that. And as we generate more data, I think we'll have a better understanding of that. And that's pretty much it.
Thank you.
Okay, thank you.
So I'm guessing you already know there is no question option. So the next one is Rebecca Pearson from Glass. She's going to talk about creating your own beach data story using the My Glass data portal. Rebecca.
Hello, again, my name's Rebecca Pearson. I'm really new to GLOSS, or the Great Lakes Observing System, this year. So I'm learning more about it, and I'm also a newbie to the Great Lakes Beach Association. This is my first conference, and my first day here, and it's been wonderful. It seems like an exciting time to be a beach manager and a researcher. So many tools out here, and I hope that what I demonstrate here today will be another tool that you can go to. And so I wanted to take a little time to describe what GLOSS is. I hope you're aware that it's out there. It's a regional organization. It's one of 11 regional associations of the Integrated Ocean Observing Network, which is an office under NOAA. And our primary mission is to enhance the availability to collect, deliver, and use ocean and Great Lakes information. So IOOS, which is that Integrated Ocean Observing Network, is a partnership of federal agencies, academic institutions, regional associations, and private sector parties. So they're all working together to try to share data that's open to the public. So in summary, we are a data aggregator, a data manager, data distributor, and we fund projects that further our mission. And Elizabeth, who's standing right next to me, will be talking at the end of my demo on a project that we've funded, which is really specifically for beach managers. Now, the GLOSS data portal is available to use for other subjects, including beach safety, fishing, boat safety. So a plethora of other issues you can explore using the data portal. So one thing I'd like to highlight is new version of our data portal has a My GLOSS feature that allows you to create your own data story. And you can do that every day if you want to. Because it allows you to have a means of saving your favorite data sets and going back to it, or exploring other data sets and saving them in a different type of story. But so I'll demo using one story, and it's the My Lakeside story. Lakeside is a beach resort on Lake Erie. It's been around for 140 years. It attracts many visitors every year. So here's a picture of Lake Erie, a map, and if you dig further, it's just above Sandusky. I think it's highlighted a little bit there. You can see the circle around there. That's Lakeside, Ohio. And here's one of the major issues is HABs, of course, which has been prevalent throughout many forums, not just the Great Lakes Beach Association, but many regional organizations are working on this topic. So here I just pulled out a screenshot from a NOAA bulletin dated August 10th of this year, and it shows you where the HABs are at that point in time. And you can see that Lakeside doesn't have, at that date, doesn't have to worry too much about HABs at that point, but you know, there's that ever-present concern. And so what I'll use the data portal is to look at that blue-green algae parameter. And you can try to look for the trends that might indicate HABs. So with my gloss, I'm going to demonstrate 6 functions: creating an account, searching and choosing data sets, exploring models with a virtual buoy function, which is really neat, graphing and comparing these data sets within the browser, and then saving those data sets. You can create a link and share it with your colleagues. So let's dive into the data portal. When you get to the page, you'll see this welcome page. It tells you in summary the major features, the data catalog, which is just a listing of all the data sets. Then you can go to the mapping feature, which will display the data sets that you are interested in. And also, once you develop your MyGloss account and pick your favorite data sets, you can create alerts that will go to your email or phone, so that when there are certain thresholds that hit, you'll get a message about them. But we're not going to look at that feature. I think we're going to run out of time to do that. So here's a view of the catalog. And what this slide shows on the upper right-hand corner, where there's like an image of a man, that's when you click on that, you can create a MyGloss account. And it's a very simple form. You fill out your name, give your email, create a username and password. And this information is not used for anything. It's just there for your account to be created within our system so that you can fully utilize all the features in MyGloss. So going back to the catalog view, you can search for different parameters. For this demo, we're taking Lakeside, Ohio, and you can run a search and pull some data sets out of that, and buoys. There's satellite imagery too as well. But for this instance, I'm picking a buoy near Lakeside called Marblehead. And so you can click on that and save it. And once I've clicked on the buoy, there's the parameters that it's sensing, and one of them is the blue-green parameter, blue-green algae. So I clicked on that, which activates a graph parameter underneath that list within the buoy window. And if you activate that, you can map out blue-green algae in a time series plot. Also plot the window, another smaller window that's active allows you to save that data set. So maybe it's not worth walking away and trying to point to where it is, but it's a smaller window on the left side. It says current collections. You can click on a little icon that looks like a new file, and you can click that on and write a new name, so you can name it something and save it for the next time you get in, or you can share those results. Again, focusing on that left side, you can see it's a little hard because I'm not demoing this in real time, but there's a little, I renamed this data set Lakeside Beach, and that's reflected underneath that small window on the left side of the screen. Another thing demonstrated on this screen slide is you can overlay data sets. And so what you see here is a data set on wind. It's a model for the entire Lake Erie. And what you can do there is you can create a virtual buoy. You select that on the left side of the navigation bar, which allows you to pick a point on the data set and then bring up a specific value for wind in that area, and that's what you see on this screen. You can also plot both types of data sets in a time series as indicated here in the window at the bottom of the screen. So you see wind, which has the little flags that turn around over time, and then the blue-green algae. Last slide for the demo. Not only can you save things and share them, you also have certain helps, and that's in the menu bar on the top right. So if you click the three little bars, you'll get a feedback form, which you can type in anything, and it goes to our data management team. So we have a whole team of people that can help solve problems. If you have a question about a data set or would like more data within that's outside of what is viewed through the data portal, we can give that data set to you. And then we have 3 tutorial videos that are really great. So if you want to go back and on your own time, watch those 3 video sets and you'll learn, you'll be an expert in our data portal. So those are, we went through 6 functions, and I hope that you can go back and get your own MyGloss account. That'd be really great if you can do that tonight as homework. And use it. I mean, it's there for your use. We also are interested in integrating new data sets into the data portal. So if you're a researcher and you're closing out a project and you really want somebody to make that data available for the public, or just have it known in a metadata record. We curate all data sets for the Great Lakes region. We'd love to have more of that populated through our websites. So with that, I'm going to let Elizabeth talk about that GLOS-supported project that does Nowcast, right?
Yep. Thank you, Becky. So I don't want to stand between you and your break that's coming up in just minutes. This will be really brief. But I want to take some time to tell you about a really interesting project that GLOS, the USGS, and the Wisconsin Sea Grant are working on that I think would be of much interest, especially to beach managers. So as Becky said, my name is Elizabeth, and I'm with the Wisconsin Sea Grant. And so the project that I'm involved with is improving beach nowcasts using web data services. And nowcasts are these real-time predictive models. And so the objective of this is to increase the use of GLOS data. So what Becky was just talking to you about, this really rich data portal, specifically within the operational nowcasts models. And so specifically what I'm going to be giving you a really brief introduction to is this program called Virtual Beach. So we have the URL for it at the bottom, but that will show up again in just a few moments. So how many of you have heard of Virtual Beach? Excellent. Okay, so this won't be too bad. So Virtual Beach. So as many of you are aware of, as it seems, Virtual Beach is this program specifically designed for beach managers for creating and developing nowcast models. And most commonly it's been used to predict E. coli levels, though it has been used in the past to look at predicting halves for beach closures. And so one of the great things about Virtual Beach is that it has, it's tied in with this other data portal called NDAT, which is very similar to what Becky just talked about, her GLOS portal. So NDAT is, like I said, built into Virtual Beach. They are meant to work together. And like GLOS portal, it has these virtual buoys where you can pull down site-specific, constantly updated data wherever you want to be modeling, so wherever your beach is. And so with NDAT, the great thing is, like I said, it's meant to work specifically with Virtual Beach, but also is publicly available online. so you can access it independently as well. So with that, like I said, it was really brief, but we're happy to field any questions if we have time for that. And okay.
Thank you.
Yeah.
We have time for 2 questions, please. Not only for her, if you have any other questions for other speakers, the two questions. Okay, thank you. Thank you. Yeah. So Shannon says you guys can come back at 3:30, right? Thank you, guys. Perfect. Thank you, sir.
Again. So our next speaker, our next session chair here, I'll bring this over to you. So our next session chair could not be with us, Claire Robinson, but she called, and we've had Donnelly Dinsmore from Wisconsin DNR stepping in to take care of the next session for us, and I'll let you introduce our speakers. Thanks.
I think I'll leave this.
Feel free to pull it down. I'll put it back.
Okay. Well, the next speaker is Bob Pratt. He's going to talk about drowning and some of the epidemics that we've had with that. And take it away, Bob.
Thank you very much. We're missing a few people, so you need to clue them in. Those scientists probably that you're working with who are like, I don't need to know about that drowning thing. I'm worried about this cyanobacteria type stuff. So please help to educate them. My name is Bob Pratt. I'm the Director of Education for the Great Lakes Surf Rescue Project. We're a nonprofit organization that goes all over the Great Lakes area talking water safety to anybody who will listen. And I'm very, very happy to be here, honored to be here, because I think this is really, really an important issue. And for most of you, it's probably something that you don't think about much, but it's something that I think you should think about a little bit more. So we're going to start off, we're going to talk about the scope of drowning. Drowning is a huge, huge problem. I come from the fire service. I was a firefighter paramedic in the city of East Lansing for 25 years. I've been a paramedic for over 30 years. And it amazed me when I started getting interested in water safety what a huge problem it is. Drowning is the leading cause of accidental death in kids 1 to 4. Let that sink in for a second. If you have children or grandchildren in the 1 to 4 range, nothing kills more of them accidentally than drowning does. Drowning is the second leading cause of accidental death in kids 14 and under. Coming from the fire service this October, if you have kids in elementary school, they're visiting a fire station or having a fire truck come to their school this week because it's Fire Prevention Week, and we put a great deal of emphasis in fire safety, and I'm not saying that that's a bad thing. In the state of Michigan, we do 5 fire drills every year in schools. We do 2 tornado drills. We do 2 defend in place drills. In anybody from Indiana here, Southern Illinois, you do earthquake drills in your schools there. Drowning will kill more people than fire, lightning, tornadoes, school shooters, and earthquakes combined. Drowning is a huge, huge problem. I have to go to my notes here because this actually changes every day. We started our organization in 2016, and since then there have been 522 fatal drownings on the 5 Great Lakes. Typically half of those happen in Lake Michigan, and typically half of those happen in the south half of Lake Michigan. So far, just this year, 85 fatal drownings on the 5 Great Lakes. Those numbers will go up because we have 4 people on life support that have been on life support for a prolonged period of time. I think the shortest one is now a month. And we have one person who drove their car into Lake Michigan in Chicago yesterday. We're waiting to see whether it was a medical emergency or whether the fatality was caused by by drowning. So it's a huge, huge problem. And again, you may be going, you know what, I'm a scientist. I know how to swim. I don't need to worry about this kind of stuff. If you're a scientist and you're taking water samples and you're going out in the water, are you wearing a life jacket? Of the 522 fatal drownings that we've had, 5 of them were wearing life jackets. The majority of them are people who never intended to get wet in the first place. So the majority of them fell out of boats, fell off of piers, were washed off of piers, somehow ended up in the water when they had never intended to get in the water. If you manage a beach or a research vessel, make sure all your people wear life jackets. It's kind of like a seatbelt. No one ever thinks that, oh, I'll just put my seatbelt on in the middle of this crash. And yet so many people think, oh, you know, if my kayak starts to tip over, I'll just grab my life jacket and put it on while I'm going. That kind of thing just doesn't happen. So since 2010, we've been keeping track. We have all of these statistics are available on our website, glsrp.org. We actually highlight all of the drownings and gather as much information on them as we can. State of Michigan alone spends millions of dollars every year trying to entice people to come to our beaches. The other Great Lakes states do the same thing, but we have this Pure Michigan campaign, and we spend a great deal of money doing this public relations kind of thing. A fatal drowning at one of your beaches will decimate your public relations. Conversely, having a rescue at one of your beaches because you had the foresight to supply simple rescue equipment on your beaches, on your structures, makes a huge difference. I was talking with Mike from the DEQ, DNR, and we've had 2 situations in Holland, Michigan, where over 20 people have been saved in a single afternoon on 2 occasions by people using throw rings. You're looking at about $50 worth of equipment right here. And if you can provide this and you can save somebody's life, you can prevent that public relations disaster, and you can leverage that information so that people are much more likely to come to your beaches. So when we talk about how to make our beaches safer, we need to look at 3 areas. One is safer people, safer children, safer adults. One is safer water, and the other is a safer response. So when we talk about safer kids, programs that educate kids about water safety. Like I said, it's Fire Prevention Week. All over the United States, we will have firefighters from all walks of volunteer departments and career departments and departments with EMS, departments without EMS, doing a great job of teaching us about fire safety. We need to do a better job teaching our kids about water safety. So consider partnering with organizations like the Red Cross. Think about partnering with organizations like your fire department. Swim lessons. If you have somebody that provides swim lessons in your community, try to work with them and do a day at the beach and invite them to come and to educate people about water safety. There's some other organizations. That's Bobber on the left. It's available from the Army Corps of Engineers. That is Josh the Otter on the right. He's from a nonprofit based out of Nebraska. Somebody shaking their head in the back, they know Josh, the Otter. There's a book that goes along with it, and a lot of Rotary clubs have available to them the costume and books and things like that. And then there's a tremendous amount of outreach stuff that you can do. If the fire department is having an open house this month at their firehouse, probably a little late this year, but next year, consider asking the Red Cross if they want to come and do it, asking your Parks Department if they want to participate and to help educate our people so that we can keep them safer around the water. Looking around the Great Lakes region as a best practices, this is Grand Haven, Michigan, following the drowning in 2003 of Andy Fox. The community created a task force, and one of the things that the task force did was to create this beach safety challenge. They bring at least 500 people to the beach, 500 teams to the beach every year and educate them in water safety. At the same time, they do some athletic type events, and they crown champions, and it is a huge event that is very widely popular. We have a booth set up there. The TV station, local TV station meteorologist comes and talks about water safety. The Coast Guard comes and does a demonstration. This is the kind of thing that we can do to educate our kids and our adults to make them safer around the water. When we talk about a safer response, how many of your staff are trained in CPR? Why aren't they? It is a 4-hour class, and if something happens, it can literally save a life. I look around the room, and a lot of us are starting to, get gray hair, get gray beards. We're not any younger. So not only can it save a life for someone that has a heart attack here, but for someone who has a drowning episode. Again, very, very, very important. Check out what your local fire department, police department, rescue squad, what they have available. You may be surprised. They may say, you know what, we don't respond to water emergencies. We're not trained to do it. A couple of really awful situations happened in California where fire departments responded, but since they weren't trained in water rescue, they sat and waited, and people had fatal drownings in water less than 5 feet deep because the fire department chose not to go out into the water. So you may be relying on your fire department thinking, oh, they'll be here and take care of the whole situation. Where they may say, we don't go into Lake Michigan. We don't go into Lake Ontario. So you need to be aware of what their possibility is. Just putting simple life-saving equipment on your beaches makes a huge, huge difference. We fought in St. Joe, Michigan for several years, and they argued that it was a liability problem, and then they solved the liability problem. It's not a liability problem. There's never been a municipality successfully sued for providing rescue equipment ever. Then it became a money problem. So the Cook nuclear power plant stepped up and said, we'll give you 10 grand or whatever you need to provide the throw rings. Within a month of the throw rings being provided, they had saved at least one life already last year. So lifeguards are the gold standard. I hope in my lifetime to see more of our beaches have lifeguards. It's something that we're really pushing for, but it's not an easy task. It's not like you can wave a magic wand, and even if the governor said, you know what, we're going to put lifeguards on all of our beaches, we don't have trained guards to man all of those stations. And putting untrained, unsupervised guards on those beaches is not an acceptable choice either. Also, talking about public events. If you have a Coast Guard station anywhere near you and you're doing an event, invite them to come. Again, invite the Red Cross to come, invite your local fire department to come. Collaborating is really something that's really, really super important. Safer water, lots of things that we can do to make the water safer. Some of them overlap with a safer response. The top right picture here is from just down the beach here in Marquette. The former Fire Chief Tom Belt, Fire Chief Tom Belt did a great job. And this is a rescue station that they set up on the beach that's got lifeguard rings and throw rings and loaner life jacket programs. We just had a life jacket used in a rescue in Muskegon, where a couple people got caught in rip currents, and we got a couple of speakers talking about the dangerous currents. I'm going to really breeze through them because my time is short. The flag system. I'm not a huge fan of the flag system because there are some inherent problems with it. One is they have to be changed. We know how fast the weather can change here in the Great Lakes. So you need to have some staff that's going to evaluate the weather, that's going to change the flags on a timely manner. And sometimes with the economy being what it is, we just simply don't have the staff in order to do that kind of thing. Call boxes. We've had great success with some of the call boxes that are put in. We've had some not so successful situations where the water level rises up and impinges on the call boxes, and they've had to be moved. So kind of pluses and minuses with all of these. The bottom right picture is some water safety stations that the Sea Grant and the Department of Natural Resources and a couple other groups have gotten together and purchased and put on some of our beaches. especially those remote area beaches where we're not going to have a lot of people that may be there, so that we can improvise rescue equipment. So the bottom right picture, you're looking at about $250 to $300 worth of equipment. That includes the rescue station. That includes the box that it comes in. Some areas in Wisconsin, Port Washington, Wisconsin, got a local, I think it was a Home Depot or Menards or one of those big box places to donate the boxes for their area. So if you have a marina in your area, they might be a source that they'll be willing to provide some of this equipment with a small label on it that says, you know, rescue equipment provided courtesy of Bob's Marina. The top left is Muskegon. They have a great task force there. Again, similar type equipment. So Grand Haven is really kind of one of the state of the art. That's the bottom left picture. If you take the throw ring off of the pier, it automatically calls 911, and there's a private beach cam that automatically pan, tilts, and zooms to the area in question, and the dispatcher can watch what's happening in real time. And we have video of and audio of the dispatcher saying, okay, there's 2 people in the water. They pulled another throw ring. Okay, both of them have throw rings. Both of them have flotation, and flotation really is the key when it comes to drowning. And they can all do this in real time, and they've had great success there with rescues. Again, some of these are right around the corner. We were just talking about Picnic Rocks, which is a very dangerous area here. If you're interested in geomorphology, like my friend Guy Meadows talks about, they have a tombolo there. If you know what that is, it's awesome. If you don't, look it up. It's really cool. But they had several people who have drowned there over the years, and so the top left picture is the sign from there. And the bottom right picture is actually a memorial for the people there. The top right is Grand Haven Beach, where they've had, again, Andy Fox and another individual drown. And then the one in Frankfurt, the bottom left, was made by the family of this young man who drowned there, and they paid for the rescue equipment to be put there. Again, this is Port Washington, Wisconsin. There's tons of signage available. Format it to the conditions at your particular beach. We're going to go through a couple of them now. These are all available from the Sea Grant and the Dangerous Currents, and I can give you the website information for all of these. That top right is that tombolo. It's a sandbar that runs all the way out to this picnic island, or Picnic Rocks, and when there's a current running, it sweeps people right off the island. Common sense would tell you, oh, you're standing in waist deep water, and I've been pushed off the island, and now I'm in, you know, water over my head. It's only that far. I should swim against it. But like our traditional rip currents, common sense may not be the best option, and actually swimming away from you is the safer option. So all of these are available. The bottom right, the structural currents. Structural currents are responsible for the majority of our incidents, both fatal and non-fatal. They also have the structural current sign reversed, so that if you happen to have a northern facing beach on the Michigan side of Lake Michigan, or a southern facing beach on the Wisconsin side of Lake Michigan, they have that sign also available there. We have this information on our website. You can use it. Please feel free. We only ask that you leave our logo on it. If you want to make our logo a little smaller, put your logo right next to it, please feel free to do that. One of the big problems is that drowning isn't like Baywatch told us all. It's all a lie. There's no waving. There's no yelling. Drowning happens very, very quickly. Educating your staff about what drowning actually look like and how to make rescues using shore-based equipment like these throw rings can go a long way toward making your beach safer. So again, just in review, safer three areas that we need to worry about with water safety, whether we're talking about backyard pools or we're talking about Lake Superior: safer swimmers, safer water, and a safer response. There's brochures that from our organization around, and I'd like to encourage all of you to check out the Great Lakes Water Safety Consortium. It's a new organization. It's just started in the last 2 years. We've done 3 conferences so far. We partner with all of these agencies and more we'd love to partner with you. It is a Great Lakes-wide program, and it's a best practices program. It was started by a communications specialist from the University of Michigan who had a non-fatal drowning in Leland, Michigan, a couple of years ago. And an hour after he was rescued and had CPR performed on him on the beach, another individual, a 15-year-old local boy named Brian Ralston, had a fatal drowning at the same beach. And Jamie Ratcliffe, our leader, is very, very passionate about water safety. And last year we did a conference in Cleveland and a conference in Grand Haven. The year before we did a conference in Michigan City, Indiana, and we are going to be doing other conferences. So thank you all very much. I went over, so I talk back.
Do we have any questions?
Given statistics around children drowning near more popular beaches, are there any junior cards during lifeguard programs to train kids?
Repeat the question.
So the question is, do we? You're not from the area, are you?
No.
Okay, so the question is about junior guards for our beaches. Unfortunately, in the state of Michigan, I'm only aware of a handful, under 5 public beaches that have lifeguards at all. Down in Indiana, it's a little bit more. Illinois is a little bit more. Wisconsin has a few. Lake Erie has a few in the Cleveland area, but the vast majority of our beaches are unprotected by guards at all, along with the junior guard program. So it's something that we're working with the guard programs to try and hopefully get a junior guard program, but we need to get guards on the beaches first.
There's dual benefits from that. One, obviously you're training the kids on how to avoid drowning. 2, these are summer camps that for 2 weeks the kids are being essentially going to a camp, getting exercise. It can also serve as a profit center for that local city or community.
Absolutely. If you go to beaches in California, not only are you protected by the lifeguards up in the stands, but many of the kids have been through a junior lifeguard program and are now high school kids or even adults, and they have that water safety background that unfortunately our kids never get. So thank you. Good question.
Thank you. One quick one.
Is that the problem with the equipment that's left unmanned, the rescue equipment?
You know, not really. There's a little bit. Are you from?
Yeah. No, in Marquette here, no, they haven't had.
So we've had some theft involved a little bit. Our suggestion is make it ugly and put a label on it that says property of Oceana Sheriff's Department. And that tends to make people do it a little. And also in the state of Michigan, we have it's a higher misdemeanor. It's more of a crime to steal rescue equipment, and it's a felony if the rescue equipment is stolen and could have been used in a rescue. And we label that on a lot of our areas, but it doesn't tend to be. Actually, real quick trivia as I'm walking out, because they're going to give me the boot here. The biggest stuff that we have is in the line, and believe it or not, it's from fishermen, and they take like this big a section at a time. I see a couple of nodding heads. You know why? They forgot their stringer. So they all they need is like 3 feet of rope, and oh well, they're not going to miss 3 feet, right? I'll be around. And brochure, yeah. So we have one from the consortium and from myself. If you need more, please see me in the back.
Thanks, Bob. Well, we're sticking with the tradition of calling in the substitutes, and the next substitute is not going to be Jesse. It's going to be Marie Tom from University of Minnesota Sea Grant.
Thank you. This isn't on, is it? Doesn't sound like it. It is? Okay. My name is Marie Toms. I'm with Minnesota Sea Grant. I have been on the job for about 5 or 6 weeks, so this is new to me. Thank you. This has been very educational. I just came from Alaska after 20 years there. Whoever spoke earlier about liking the snow here and the cold, I was here for 7 years and decided I wanted more of both. So I went to Fairbanks where my first 3 winters were 55 below zero, and for the chemists in the room, I forgot that propane liquefies at that temperature. It explains why a wood stove is a good thing, otherwise you never have coffee. So Alaska is thawing, and I moved back where it's going to hopefully get cold again. So I hope you'll grant me a little bit of leeway. I got the material to make this presentation not too long ago, so I have been educating myself as quick as I can, and I was lucky enough to sit this afternoon with Dr. Wu and Todd to get a little bit of an update on what we're talking about. So Jesse Schomburg, who was to make this presentation, asks you to think of something as we go through these slides. And what he wants you to do is to think about how you and your area of expertise and the Great Lakes Beach Association might work together with the folks mentioned here in this presentation to improve beach safety. And from what I'm gathering, and especially from Bob's last presentation, several of you are already kind of well along that way. The Coastal Storms Program is funded by the National Oceanic and Atmospheric Program, and this is an initiative that brings together organizations from all sectors to a specific region, which should be clear.
We're going to get you the mic because we're on the web.
Sorry. Which should be clear given the other presentations. Each initiative lasts from 3 to 5 years and seeks to bring additional human resources, focus, and funding towards these goals. And the goals here are to prevent, and I realize what I've done here, sorry. There we go. And the program goals are to prevent loss of life and property from coastal storms, to lessen the negative economic impacts on Great Lakes communities and businesses, and to sustain the natural environment. And while the focus of this program has rotated around the country, each region gets to kind of tailor this program to their own needs. My understanding is that the program has funded 10 projects in the Great Lakes areas relating primarily to beach hazards, resilience, green infrastructure, and stormwater. And as Bob's presentation made perfectly clear, I thought, for the Great Lakes, the goal of preventing loss of life was met in part by focusing on efforts to reduce drowning, especially in our case caused by dangerous currents. So the topics for today will focus on 3 different projects with new outcomes for this year. There are 2 projects, one led by Guy Meadows, who you heard Bob mention, and one led by Chin Wu, also including Todd, Jean, and Jesse, and I'm sorry I forgot Jean's last name. And that project is focused on better understanding dangerous currents in the Great Lakes towards improving recommendations for beach and water safety and increasing the ability to successfully predict and forecast rip currents. Both Todd and Dr. Wu's presentations after mine will go into greater detail on both of those things. The third project, again, thank you, Bob, was designed to increase the number of Great Lake beaches implementing best practices that reduce drownings and dangerous currents and to help to create effective beach safety messages. And the final outgrowth of this was to foster the coordination of the Great Lakes Water Safety Consortium. This was formed in 2005, and Bob gave quite a good definition of what that is. This slide introduces Guy Meadows' project on that focused on mapping resources, data collection, and management on dangerous currents field research. Now, Jesse sent along this slide to say, to measure currents, you need 3 things. You need an observer, you need a floating object or a drifter, and you need a timing device. One of the activities in the projects that we're covering here was completed with the release of drifters at 2 beaches on particularly wavy days. And the picture on the right shows you a dry bag with the GPS receiver in it, which is the whole thing together called a drifter. And to give you an idea of what those drifters tell us, Jesse thought it would be helpful to have a sense of how fast things move in water, and he picked Michael Phelps, who can move at apparently 2.1 meters per second last he checked. And Jesse went to the local YMCA, looked at the speeds of some local swimmers in his kids' team, and found that more or less a 13 to 14-year-old boy swimming about 50 yards does that in about 1.95 meters per second. So keep in mind that Phelps goes at about 2.1 meters per second and a team does about 1.95. Looking at Holland State Park Beach in Michigan, this project placed drifters at the beach without known rip currents, and the drifters traveled along the shore. The top drifter speeds were about 3 meters per second, so a little bit faster than Michael Phelps, The average drifter speeds were much slower, about 0.5 meter per second and slower still. And there's Michael Phelps. Also at Grand Haven State Park, the project included bathymetry 3 days apart, one pre and one post-storm. On the left, you can see the pre-storm bathymetry. On the right is the post. The darker the blue, the deeper the water. When looking from the left-hand image to the right, note that the dark blue holes near shore end up filled in on the right side. Also note that the reordering of the sand forms almost a whole new beach water line, and at least 2 possible rip current channels are identified by the red circles, which I can't see from here. I don't know if you can see on the right side one. One of Jesse's points to me was that there have been questions as to whether rip currents tend to stay in the same place in the Great Lakes beaches. And one of the takeaways from this image indicates that if sand reordering happens this quickly, then predictions of rip current locations will be difficult, as these locations are expected to change rapidly. And also, areas with previously known rip channels are likely to change. So staying with Grand Haven and with Phelps a little bit, 2 drifters were released in areas with known rip currents. One drifter is shown as a red dotted line on the graphic, the other is the green dotted line. Although both drifters were carried quickly off to shore and their maximum velocity well exceeded Phelps' speed, again 2.1 meters per second, their average speed was much slower. Interestingly, the drifters ended up going very different distances offshore, and both eventually returned to shore, but it took about an hour. And looking at all the advisory materials about, float until you're out of the rip current, I don't know about you, but I'm a sinker. I don't think I could float for an hour. That'd be an awfully long time to be out there. The takeaways from this project are that almost all the Great Lakes sand beaches have dangerous nearshore currents, that they develop rapidly with increasing wave height. The nearshore bottom is continually readjusting to waves and currents, and these rip channels migrate down the beach. And the dangerous nearshore currents persist long after the waves subside. The info system, led by Dr. Wu, includes 2 components. One is a stereo camera system which can detect and measure surface currents in real time, and the other is a wave sensor installed near the shore along the beach. Both systems feed their data in real time to a website where additional processing takes place. Coupling these currents with nearshore wave height data, which from what Jesse indicates is relatively uncommon in the Great Lakes, is thought to help pinpoint what conditions are necessary for rip current development. And I think I'm ahead of myself by a slide. So this image on the right is the result of the results of a stereo camera system showing surface water movement on a high wave day. And this image shows your wave height. The third project focused on addressing dangerous currents in the Great Lakes by applying best practices in these areas and the development of consistent materials and messages for use across the region. The broad goals of this project were to facilitate regional collaboration, purchase the beach safety equipment in bulk and distribute it across the region, as Bob was emphasizing with his rings, develop consistent messages to be used across the Great Lakes, and finally to develop a plan to ensure that this can continue into the future. The objectives of the program were to develop and expand working groups, and you'll see a slide a little bit later on that talk about some of the ways in which you might be able to participate with these groups of folks, to apply the consistent messaging and outreach materials and distribute the beach safety equipment. Starting in 2014, individual state working groups were formed to figure out which beaches to target and determine equipment needs for each. And in 2015, a series of videos, graphics, and messages were developed for a Great Lakes-wide social media campaign. If you Google #CurrentsMart, you shall see that information. And this year, project efforts have focused on final distribution of safety equipment to promote their use, as well as part of a sustainability plan. Part of that plan was the formation of the Great Lakes Water Safety Consortium. This will look very similar to the slide you saw before. These are a few examples of the types of rescue station installations that were supported by this project. And again, what Bob said about stenciling the buoys was something like Sheriff's Office seemed to indeed work to prevent theft. And as he also said, the equipment at each of the stations was selected by the local beach managers. Where I'm at in Duluth, for example, the fire department officials didn't want to encourage people to go out in the water themselves, so they did not provide life jackets with those stations. And also, interestingly, Jesse indicated that he knows that bystanders are eager to help if they know someone's in danger. And apparently in Duluth, as public awareness of rip current awareness and safety has increased since this campaign, by the time the fire department gets to a beach for a rescue, there's already been somebody on the beach who's attempted the rescue and has been successful. So the public outreach and education component seems to be valuable.
There's a volunteer life-saving service in Duluth at Park Point.
Excellent.
Great program.
Making sure that high-quality equipment is available can reduce the risk of rescuers needing to be rescued themselves, as I think we've all pointed out. More of the placards, again, as Bob said, that you can tailor to your specific needs at your specific beach. Here's some of the numbers for the water safety equipment and signage that's been used. Some of the equipment items that are at these stations include throw bags and rescue cans, rescue tubes and whistles. And I'm going to skip some of this other stuff because you've covered that well. A couple of quotes from folks who are appreciative of the equipment and the impact that it had on them and others. The last call to action, and one of the most important outgrowths of the Best Practices Project, was the formation of this Great Lakes Water Safety Consortium. And the hope is that the consortium will be a way for folks here at the Great Lakes Beach Association and you as participants to continue some of these public education efforts going on into the future. This consortium, again, just to reiterate, is a community of best practice people. They bring together first responders, community leaders, park rangers, research scientists, lifeguards, meteorologists, survivors, loved ones, and other water safety advocates. The goal, of course, is to end drownings in all of the Great Lakes. Another slide of all of the participants who are part of that project. This is one of the brochures. Again, some of the graphics will look familiar. I'm a product of that endeavor as well, distributed at Parks conference material. And to come back to what Jesse asked at the beginning, that you think about ways in which you and your expertise might be involved with this effort. These are the committees that are part of the Great Lakes Water Safety Consortium. We would hope that if you have interest or some of these appeal to you, that you make contact with these folks here, Jamie, to express your interest and see what you can do. And last but not least, everyone here today has mentioned something about communicating their message to the public and trying to figure out how we get people to pay attention to things they're not maybe necessarily inclined to be interested in. And I don't know about you, but I never really thought about looking for bacteria information when I go to the beach. I usually want to know what the weather is, and I just recently learned how to surf, so I care about surf heights, and I'm following my surf app. But you made me think of a lot of questions I really should be asking now. And there was an Intercept survey that was done for audiences on Milwaukee and Duluth beaches, and predictably they said we really care about weather and temperature. And to kind of address that need, if you can co-locate the stuff you want them to know with the things that they already want to know, you're more likely to get them to take their medicine with a little bit of sugar, as it were, and convey those things that you think that are most important for them to know. Both the Great Lakes Water Safety Consortium and the Great Lakes Beach Association have as their center, it appears to me at least, a desire to keep beach users safe. And hopefully we can all figure out a way to kind of work together to figure out how we can do that better. Again, thank you for your patience with my somewhat herky presentation here. If you have questions, I'm interested in hearing what you want to know, and also would give that back to Jesse. Thank you.
So are there any questions? So our next speaker is Todd Bryby from the Wisconsin Oh, Chin Woo is going to go first?
So Todd asked me about, because I'm scared of the audience, I do not want to be the last one. So I decided to be earlier one. Sorry.
Okay. Well.
So scared. No, I'm joking. It will be much smoother when we talk about the influence. After that, there are certain things that the particular outreach, education, and all the action that Bob and Todd and cultural management, as well as so many of you, I think it's we will move much smoother. So I have take a bit switch.
Okay.
And hopefully you are okay with that.
I'm totally okay with that. You can see your slides here.
Perfect.
Is that it?
Nope. This one. Excellent. Good. So I'm running if I need to go into it. Okay. That's okay. Okay, so first of all, I really like this picture. I really do not know where I grabbed it from. I grabbed it from the great organization, Great Lake Beach Association. Very nice picture. I couldn't believe, oh, so nice. We are talking about water quality today. No, we are talking about the safety, including water quality, as well as water quantity. Quantity of what? Like this kind of situation. You probably know this is what? San Diego. No, this is at Duluth. Very beautiful place. Look like San Diego. You look at, oh, no, I'm not looking at the background. I'm looking at on the water, what exactly called dangerous current or rip current? So-called rip current is, the so-called rip current is that you have a current that going back, somehow pull the people back. And I believe that you know. That's okay. You know it. My voice is loud enough, right? Okay. So I have to acknowledge all the people, and indeed you will see so many people. I really cannot put all the people together. I would say at least 20 people. This is not a single PI project. The difference between the Great Lakes people is that we are great. We work together. Okay? So with that said, though, I'm going to talk about the infos, and you will say, what is exactly info? Just ignore what I'm saying, info. So look at exactly what we are looking for. Bob did mention about quite a lot of statistics. I just give you the whole summary statistics, okay? Yeah, close to 40% of the deaths related to the rip current. This is very high, and I couldn't believe it's so high. It will drive me nuts not to lower down this number. Okay? We need to do this, not by Bob, but all together. Okay? Of course, Bob, you are the man who rescue all the people. Okay? Of course, hopefully you will never rescue anyone. We are all in the same place. So with that said, though, you know that we would like to have a lifeguard, but it's very unlikely. It's very unlikely. You will say, well, why is it very unlikely? Because we are so rich nowadays. Every beach can go to have a lifeguard. It's not quite true. The lifeguard cannot do 24/7. Indeed, quite a lot of time that we found the drawing is what? In the early morning. The kids wake up early. Oh, I want to have fun. That causes trouble. Okay. So with that said, though, yeah, you know, flag system also indeed what? If you have it's probably what? If you have a static signage or flag system, you probably want to take it out. That has a psychological survey showing that if you have that without changing, people will ignore it. Just take it out, please. Don't even use it because then people is, oh, I'm not sure. Versus what? If you never change it or change it very slowly, people ignore it. Yeah, no big deal. Okay? So with that said, though, there are a couple of things that what? Really prompt us to the one that very, very important practical aspect. What do we need to do? We would like to have the system we call nowcast. So called nowcast means what? Measure it exactly, real time. When I say real time, it's called real time. I mean it. Okay? It's not 2 or 3 hours or 10 minutes. No, real time. When you look at your phone, iPhone, you will see it right away. Called real time. Okay? And I would say it's very challenging for the people in the little bit federal level. As you look at it later, you will see that. We also would like to enhance the public recurrent awareness. Bob, Marie, we're going to talk about this. Okay? With what? A lot of people. Okay? Cannot be single organization. A lot of people. Great safety organization, consortium, right? Water safety consortium. We need to all work together. Now, I was, what's happening? Here is the last one. Indeed, quite a lot of this kind of system. Oh, we are doing it. Usually end up with what? What is the sustainability? Because we have funding for 2 or 3 years. After the funding ends, the project disappears. This is ridiculous. We waste the public dollar, tax dollar money. We cannot do that. All the things that we are going to develop is going to the federal NOAA station. It will be 24/7. They will not stop unless you want to fire all the NOAA people. Unlikely. Because whether everyone wants to know whether, regardless of which party you stand apart, we all like weather. Okay, weather information. So with that said, though, it's our goal. Now, I'm going to tell you a little bit about what is the characteristic. Oh, it's blocking it. The word called characteristic. Well, give us some kind of information. Most of, I use a most of the big current or dangerous current always what? Once you see the wave breaking, look at it. There's a dangerous condition. Most of it may not be exactly true. Okay? Close to 0.3 or 2, usually. And you say, who cares about 0.3? Imagine you are swimming 2 meters forever? No way. 0.3, oh, then you get exhausted. Usually you get exhausted. Oh, my muscle pain. Okay, this cause the width in the Great Lakes, 5 to 50 meters, very, very narrow. Totally different from Mega, from the California, from the Gulf. It's different. We are just different. Okay? Our rip current is totally different. The scale changing 5 to 30 minutes disappeared. Very short. Okay? With that said though, there are a couple of mechanisms that if you look at that animations, hopefully it work. If it's not, you can ignore it. Definitely what code is not available. Fine. But overall this way, you do have a lot of offshore bar. What's an offshore bar? You have a sandbar. Sandbar is not always continuous. You can imagine what is the sandbar is what? Sandbar, the wave break. Eventually the water level here is higher, eventually we're going back. One of the mechanisms, sandbar. Okay? It's very, very common, this kind of thing, particularly what in the sandbar area. People like to go there. Now the other one, oh, you see the video, structure induced. You put the dye there, you will see it. Oh, jeez. Okay? The next one, I really want you to take a look at this. This is the most important thing that I think doesn't work. Okay? Doesn't work. In the computer, different. Okay? This is what I call so-called meteor tsunami. Now, you probably heard a lot of reef tides. Do we have a tide in Great Lakes? Little bit, small. Do we have a stations? Yes. We do. So in the Great Lakes, we have the reef stations, the first terminology in the world, reef stations. We ignore that. Unfortunately, this video doesn't show. You will see that, oh, the people just speak and drums. Oh, a lot of fun going out there. This is the one being ignored, and this is one of the most dangerous ones. The people going there, there's a couple. Unfortunately, I cannot see it. Unless you can, Matt, you can fix the video if you could. That would be great. Okay? You want to see the video? With that, we have a couple sites. I'm not going to repeat the much detail. Couple sites. The Park Point Beach. Why? If you pay attention to, you see how many sandbar here? Oh, tons of it. Every place is all the dangerous stuff. Okay? Second, McKinley Beach. If you ever been there, what is that all about? People like Pocket Beach. Very nice. Pocket Beach. Eventually we're calling up Pocket Beach. Look nice, very dangerous. Okay? Third, of course, say Port Washington. Why we chose Portland? Because there was a day in 2012, I happened to be there. I'm doing something else. Okay, I happen to be there, and there was a kid, Tyler. I don't want to say that. Okay, Tyler. Oh, John, I said, oh, what happened? And indeed, we measured exactly the dangerous current. The problem in a second is that no matter what, you got to build this system regardless whatever. So we started to build in 2013, 914. Eventually, the coastal zone program comes. Okay. So with that said, though, I'm going to focus on today. There's so many. Focus on particular on Port Washington and the loop. I kind of switch it back for you to look at it. Definitely, the system look like this. Install is a 9. Oh, look like there's a 2 camera, right? Look at 2 camera. In general, look at that. This is the day. Okay? You say, well, who cares? We have all, yeah, camera now is pretty cheap. Okay? I do not believe organization cannot afford the camera. Okay? You can record the camera, but the component is not record the camera. You want to go beyond to say, well, can you go beyond to get that what? You want to check the water surface. You want to check the objects. We said that we are doing the what? Interdisciplinary. Water quality, water quantity, computer science, computer technology. We should put all together called imaging tracking. This technique that what has been done in other fields, but have not applied to the what? In our recurrent fields. Ridiculous. We should apply. We've done it. The algorithms are very simple. I can send it to you. You can just apply it right away. And you will know whether it's true, is it recurrent or not. Okay? You will get the current, you will get the speed, the direction too. And flip it back to go to the loops. The 2 camera now, and you can do quite a lot of things and say, oh, look at this. Oh, indeed, why did you put the 2 camera there? You can make it become wide angle, stitching together. Computer technology, I don't have to do anything. I ask my students and say, oh, you can take classes. They say, okay, computer technology, oh, earn a big buck. Oh, they can work on the part. Get the job done and send you the code, you can get the job done. And that's how you see the what? You can stitch it together. Then what? It's wide angle now. Are you going to afford 1 camera or what? You can put all the cameras stitching together, the whole beach's area. And with that said, though, you call wait, don't come, recurrent. I don't want you to come. So wait. Wide angle imaging tracking system. The recurrent you cannot come. Before I tell you this is not going to happen. This technology should be used at what? Real time. Okay? With that said, we also have the real-time wave gauges. It's called altimeters. Okay, it turned out to be what? A quality sensor. Most of the things that people say, oh, it's very expensive. I say, no, actually, it's not cheap. When you look around, you will find this kind of sensor is very affordable. Okay, it's so cheap that I couldn't believe how cheap it is. But most of the people will say, oh, very expensive. It's not cheap. We work together. We can implement this kind of system. If I am able to connect it to the real time so that you can see it, if I'm not, Matt, you have to help. You cannot say no this time. You cannot say no. Oh, real time. See? Okay, and now if you are trying to look for it, don't remember the address. I forget about everything. Just INFOX space the loops. Google, first one. Because it has been searching 30,000 times since we deployed in 1 month. Are you kidding me? Okay? You will say, well, hold on. What is original imaging? You can look at the imaging. The imaging that allow people who, oh, gee, what happened? Don't like to see that. What happened? What is that? Matt, you have to help me out. Yeah. I don't like that. Yeah. It doesn't make sense to me. Yeah. This computer is dangerous. Current. Oh, you can do it in the real time here too. Okay? So in other words, everything that you track it, and of course, who track it? For the people like us, you can, okay, look at it. Okay, everything and what? Send it to a file department. It's a very, very cheap cost. One-time fee. Okay? So with that, To do this one is that if you can record it and you quickly grabbing it, okay? It seems like imaging is a computer read, okay, or public bandwidth, okay? So you can also get a what? Surface temperature. You can also get a bottom temperature, okay? Because not only report the recurrent or current or wave height is important, you also want the temperature. You want to know what is the temperature. Temperature on the surface and the bottom is different. You need to know about it, okay? So with that said, if you recall, we need to do what? Can get the what? Get the up nowcast. Nowcast is now what? Casting what? Get real time. We cannot. Everything is real time. Only 10 seconds difference. Okay? So you are able to see the technology is here. The technology is now. Our kids, our young generation are so good about and do this, make it work. Is what? Are we going to work together to let them to say, hey, they are going to implement it for us? Okay? We also put this kind of thing that what, for the nearshore wave, because all the wave modeling so far at the Great Lake, at the federal, now you do the forecast. There is no nearshore wave data. Sorry. Zero data. This is the first data set that they are going to use. Oh, they are using it. We work very closely to implement it. But what we have not done is what? Forecast. You see, well, what is the forecast? We also like to know about for the next 2 hours, next hour, what is going to happen. Roughly, only roughly. You want to know roughly. You cannot say, it's going to happen. I don't believe that. The weather changing in a second. Okay? So with that said, though, we take this whole real-time information, including the NOAA information. We got to work with a federal partner together, and we are a team. All between, we talk about almost every day. Go every day for what? We say, oh, for this project, since the day one I'm in Wisconsin, in Great Lakes, 25 years. We work together. We never stop anything. Okay? Work together, eventually come up with what? There's one issue here. A moment ago, I said that for the recurrence, what is the size? I mean, you know the whole forecasting system, you know what is the size of the bridge? 1 kilometer. Are you kidding me? Find me the 1 kilometer. You say, oh, can you do that? You will say, no, this is local. This is local. This is your issue. We cannot deal with it. Too expensive. Don't worry. Be happy. Right? We need to do this. There is a technique out there called seamless mesh. We nested it in the detail, take all the federal data, and then put it to the local, become the detail. Detail can go to what? Our resolution now can go to the 5 centimeter if you want. Computing is no problem. Because we are running the whole Great Lakes. We're running the what? Local pitch. That's all we get. Okay? Now, the system itself is called seamless mesh automatic regrading techniques. I call SMART. It's smartly integrated, and so that you can have a high performance fidelity computing to capture the big curve. That's what we look for. Okay? With that said, though, you see how good it is? I say, it got to be good. Otherwise, I will not present it. Okay? But this is a joke. We need to be good so that we can capture that big curve. Okay? Now, for what? Yeah, detail here, I'm going to talk about it. So a moment ago, I saw that what? Structure. You know, oh, where is there a wing structure coming from here? We have identified, always on the lee side, there was always another big current at the channel. No one talk about it. No one talk about it. They say, oh, we're coming from here, people are here. No, most of people say, yeah, this is calm area. Actually, it's not true. Big current is what? Very, very what? Strong there, as you can see on this one. And we measure it, we modeling, we always see the real time. Okay? So this one is a new education that we need to tell people about. Does not mean you are on the lee side. You are not worried about structural induced current. This is the real time, of course. I'm going to try one more time. Again, you cannot say no. If it doesn't work, whatever. But overall, you will see this kind of system all in the real time, like this way. Okay, you can see this one in the real time. You get all the thing what? You can get it in a second. Now, if you go to the website, you can download on iPad as well as you can what? Use a Android. It's all in real time. Most of people use a smartphone. So to close this one, we definitely what? Make it what? Wait. And if you are looking for the technology like this, talk to us. Us, not me only, talk to us. We should all work together. Make this become very affordable. We can make it if we work together. Okay? Water quality, water quantity need to be all together. Because that's what the spirit of your first picture. I like it. Okay? With that said, the real-time wave is there, the technology is there. Smart is also developing. You can expand it to 101 and 1. Who run it? Local police station. We set it up, they run it. They take it back from the federal, they are not going to go to the local. Okay? And increase what experience about at what condition. It's called expert opinion. Like Bob, oh, I really know what's going on. Write it down. Okay? All this experience need to accumulate. With that said, though, I said that what? Info technology definitely is there. Now, how are you going to take it to the next step? Is what we believe. I think the most difficult thing is what? Engineering, I found is the most easier. Input, output. Okay? The most complicated way is what? Science. What science? Social. Politics. Education. This is the most difficult. So I leave this top topic to what? Top. Because I was so scared. I said, I don't want to talk about it at the end. So he is going to what? Excel to this topic, going to the next mode. I said, before I end, we need to work together to reduce this 40% of the rip current to be what? 0. Not only that though, Great Lakes, are you kidding me? All of the United States. No, all the world. People in the Indonesia or the third world country, they say, oh, nice. Oh, we need to go to everywhere to tell them the technology is here because we are the people in the Great Lakes. We are the great people. Okay? Good.
Thank you, Chin. We got time for one question?
Okay, chat about any time. Okay?
Todd?
Move forward. Okay? I'm supposed to follow this.
Yeah.
It was my idea for Shin to go first.
Yeah, well, you know,
Please, Jen.
he steals the scene a lot.
Absolutely.
This is Todd Riley with Wisconsin Sea Grant.
Wisconsin Coastal Management.
That's
okay,
Coastal
absolutely.
Management.
Wisconsin Sea Grant is a great outfit as well, too.
Yeah.
How do I follow Chin? What is the definition of luposlipophobia? It's my favorite far side. Any guesses? The danger of running around a kitchen table on a newly waxed floor in your socks while being chased by the consumer.
Microphone. Does this work? Yeah. Okay, perfect.
For now.
For
now. So my name is Todd Riley. I'm with Wisconsin Coastal Management Program, and I'm out of the Department of Administration, and I'm based out of Madison. And I am following up with Chin on the second portion of the Infos project. And I do want to put a plug in as well. Bob has said it, Marie, and Chin. Kind of taking off my water quality hat and putting on my dangerous currents hat. My working relationship with GLBA has been exceptional. It's a great organization, a great group of people. And for the past couple of years, you guys have opened up the association's conference to really discuss and talk more about dangerous currents and their impact. And in Wisconsin, we've been not only trying to focus on water quality as we continue moving forward, but we've also been incorporating hazards and dangerous currents at beaches as well too. So I'm hoping regionally that the different groups can work together more closely and continue to promote dangerous currents. So with that, Bob touched on this a little bit as well as Chin, but obviously as a background, dangerous currents are a persistent hazard. The number of fatalities and rescues that have been recorded since 2002 have been exceptionally high and too high. Public awareness in general about rip currents is quite low, something that we've been finding over the past couple of years as well. Outreach events that we've had in the past year have really demonstrated to us that there's really not a broad understanding of rip currents, both that they exist in the Great Lakes, how to address them if you find yourself in a rip current or someone else near you is in a rip current. So the awareness, and I think the work that Bob has been doing has been exceptional. I think it's because of their group that we're as far along as we are, and we have been. I think the project that I'm working with Chin on with this, I think also addresses gaps in understanding of rip currents, how rip currents work, how they act, and the ability to nowcast and forecast them as well too. Bob and I were talking before about inconsistency and how resources are provided across the Great Lakes when it comes to education and equipment and signage and just general resources that are needed to address this. Certain groups are more susceptible, and I'll touch base on that. And Bob actually also highlighted as well too that the majority of the Great Lakes beaches do not have lifeguards. And talking to my colleagues in Wisconsin and other coastal communities, there's been even cuts on lifeguards within the past couple of years as far as beaches themselves. So it's a persistent and continuous problem that should be addressed. So Marie touched base on this. So there was a 2010-2011 GLRI funded project that looked at beach user surveys in Duluth and Milwaukee, kind of gauge user information, user understanding and awareness and needs. The other foundation that leads to the work that went into implementing dangerous currents or implementing best management practices and the project with Infos as far as what coastal storms have been funding through NOAA were two studies that were done. The one on the right was funded and worked through between NOAA, DEQ here in Michigan, and Sea Grant. The one on the left here is a Great Lakes Beach Hazard Developing Risk Communication Strategy. That was actually funded by NOAA Coastal Storms Program that went to Eastern Research Group, and it did a couple different things. It actually took a look at identifying perspectives that beach users have about hazards and risks at beaches. So kind of trying to quantify and collect that data. It actually evaluated the messaging and it evaluated the delivery mechanisms of the programs that were out there to increase awareness. It also made recommendations as well as what kind of messaging, what kind of delivery mechanisms need to be in place in the Great Lakes to lower the number of fatalities and rescues. And also really looked at the targeted demographics of who's impacted. And on the bottom I have the three key audiences that are impacted most by rip currents. Youth, primarily males, but youth too. Tourists and parents of small children. If you look at the incident databases that the National Weather Service has, that's you can link to through dangerouscurrents.org. You can not only look at fatalities and rescues, but if you delve a little bit further and talk to Megan Dotson, who's with the National Weather Service, you know, she's talked about the fact that when it comes to tourists, there are incidences where a lot of people, I think, about 90 miles away, so tourists are coming in from other locations to visit North Beach in Racine, Bradford Beach in Milwaukee, for example, that there is this distance or geographical aspect to those people traveling to coastal beaches. And so a lot of this lays the foundation for how we approach education communication with coastal communities and user groups. This, again, Chin kind of highlighted this, but you know, obviously the overall Infos project is looking at doing some nowcasting, forecasting to detect rip currents. There is the communication portion, capacity building portion with communities as well as education, which we'll touch on, and that is the list of project partners. Throughout this presentation, I realize that I'm the barrier to fresh air, food, and beverages, so I'll keep this short. But the take-home messages would be partnerships. This project would not be possible without partnerships. Identifying community needs, which goes into really working with the local communities and stakeholders and bringing them into the process and bringing them into working with us on the project elements. Building local capacity and community resiliency as well too. So resiliency, you know, is everything. It's infrastructure, it's dangerous currents, it's water quality, it's all that collectively. And so we're really looking to try to build that capacity within local communities to kind of address that resiliency. Sustainability, Chin brought this up. How do we keep this project sustainable in the long term? You know, this is looking at developing a cost-effective technology or method to actually address this hazard. How do we keep it cost-effective? How do we keep it sustainable? Improving public safety and increasing awareness. You saw this map, similar map, so these are the three locations. I'll primarily touch on Port Washington and a little bit on Duluth. Milwaukee will be making more strides with that site next year. So you saw the Infos website, so that's a portal for the data that's being brought in, and you saw the Infos website for Port Washington and Duluth. One thing I do want to mention too is when it comes to the coastal communities, I should mention as well too, with their involvement, if you kind of break down their involvement, their involvement has increased the impact to the public successfully, and it's increased the opportunities for eventual sustainability. If you look at what they've contributed to this project, it's identifying community needs, it is reviewing project elements, it's actually looking at reviewing project sites where the equipment will be deployed, it's assisting with preparations of equipment deployment, it is co-leading on education outreach, and it's also actually kind of helping to review the data that's coming in to ensure that there is useful data that they can use to reach out to their stakeholders and the general public and their targeted audiences. And this is kind of an example of that. So in 2015, Chin and his graduate students, through discussions that we've had in Port Washington, ended up putting together, so you've got the Infos website. What they were able to do is there's a lot of information in that Infos website to digest, and what the city of Port Washington wanted to do is they wanted to summarize that. So John Doe, Jane Doe, when they actually log in remotely through their app, through their tablets, through their laptop, and they wanted to actually go and take a look at current conditions. Suggestions that came in from the community meetings and the planning meetings that we had was something this, and this is a risk watch tool for the Port Washington study site, and it's a summarized portal to the information that would be necessary for them to make a decision. So you've got current risk levels, low risk, moderate, high risk, all color coded. You got everything from wave height and the wave period down to water temperature at the surface and bottom and everything else in between. This shot is of the webcam of the camera, I'm sorry, that is on top of the wastewater treatment plant in Port Washington that collects the observation data. The wave sensors are off the shore there. And so this is pretty much what folks would need to assess whether or not they want to take their family to the North Beach site, whether or not they wanted to actually come in and drive in from Grafton or another location west of Port Washington if they want to drive in those 30 minutes to actually go to North Beach. Another output from these planning meetings was the opportunity to look at the fact that if you look at the tourist data, people coming in from outside the region coming to visit these beaches, or if you're a resident of Port Washington and you want to take your kids to the beach after they get off of school or after they're out of summer school or an activity, there was information when it comes to wave and currents that were actually forecasted out. So you could actually look at what could be potential conditions down the road, 3 hours afterwards, 6 hours down the road. So provided that relevant information so that the public can make an informed decision. In 2015, we kind of summarized the page views and the number of visitors that we received at the Infos website. So that's the traffic. We did we worked with the City of Port Washington, UWC grant. We put out press releases. We followed up with interviews, anything we could do to actually get the word out there about this new technology, new tool that was being deployed at the City of Port Washington. And so that helped kind of drive some of it, but it was kind of the beginning, the 2015 season was kind of the beginning of actually getting some outreach out there and some press out there to start talking about this subject, talking about this tool, and talking about what the City of Port Washington is doing, and actually really what the federal, state, and local stakeholders are doing collectively to address this issue. And this is in 2016. This same risk watch tool was developed for the Duluth website, and some differences you see some of the same information, current risk levels, appropriate parameters up there with wave height all the way through the wave temperature at the bottom, current conditions. They also actually added in velocity arrows at Park Point Beach. So some additional information, a lot of that was based on, again, feedback and needs from the community there in Duluth. So again, an opportunity to tweak and manipulate the information coming in to fit what the community needs and what they want, what they think would be most effective for addressing rescues and municipalities. 2016, this is a summary of the web traffic from Infos. This we actually have a full season, May to September, both paid visits and numbers of visitors. In May and early June is when we really broadcast again, press releases, outreach opportunities. We really pushed for those opportunities for interviews, etc. And this again was co-led with the City of Port Washington and the other project partners as well too. On the lower corner, you did hear about that from Marie. It's the parkpointbeach.org website, and that is a product of what I mentioned, the 2010-2011 GLRI funded project, and that is the website that collects and displays and provides information on water quality, advisories, hazards, dangerous currents, and other relevant information. It's a one-stop shop. That works really well for Minnesota. They don't have beaches further up the North Shore, so you're really looking at Duluth as really the primary site, the primary municipality. You look at Wisconsin, we've got another issue. We've got over 190 public access beach sites in the state along Lake Superior and Lake Michigan and the Bay of Green Bay. So how do we actually kind of team and put that information together that includes water quality, that includes dangerous currents and all the various advisories that are needed. Donnelly and I have talked, and we've talked about that issue. Wisconsin obviously has a wonderful Wisconsin Beach Health website, but what are the opportunities for maybe kind of streamlining and bringing these things together just for public consumption? Leveraging opportunities. This has definitely been an opportunity, this project. We've been able to leverage other things going on. Port Washington, so Chin mentioned that in 2012, there was a teenager that had passed away due to a rip current who had drowned. From that incident, City of Port Washington convened a water safety committee that has been very active. They've been putting out beach safety equipment, they've been doing education, they've been working with schools, and so they've been really, really active in the community. We've been able to tap into that energy and those resources and those people. Twin Ports current work group as well too. I know Bob mentioned that Duluth has a wonderful group of stakeholders up there. Again, same process, a very active group. Ozaukee County outreach, so we've really targeted opportunities that when we're out and about in Ozaukee County, where City of Port Washington is located, if there's been talk and trips and tours to talk to a variety of stakeholders about shoreline issues and waterfront redevelopment in Port Washington and everything that's being invested by NOAA and the state and the local community, we also talk about the rip current projects. We talk about what the city is doing to address that in partnership with UW-Madison, Wisconsin Coastal Management, UW-Sea Grant, and everyone else. Maria obviously talked about the NOAA Coastal Storm Program Implementing Dangerous Currents Best Practices project. That's one that I've been a part of. And that was an opportunity that we took the beach safety equipment, we took the education, the social media that was developed through that project, and actually were able to assist and complement that with the Infos project. And so we had the entire Ozaukee County targeted for social media outreach every summer for the past 2 years on how to address and deal with rip current issues. So we've been able to take those other federal resources and Great Lakes wide and target specifically to Ozaukee County. Recently, I know Kim's in the back and Donnelly is here as well and Sarah's here. We put together again, Wisconsin's had a wonderful history of beach stakeholders working together in Wisconsin, much like GLBA. And so we've kind of formalized a little bit more into Wisconsin Coastal Beaches Working Group. And that's been a good process where traditionally it's been focused on research, management, and education when it comes to restoration and water quality issues. But the reception of this group that we put together has been exceptional and actually incorporating wanting to address and tackle actually dangerous currents. So it's a group, I think, that has shown that it is possible to address multiple issues and multiple hazards at beach sites. Great Lakes Water Safety Consortium, Bob and Marie have mentioned as well too, we're using that as an opportunity to target information in Duluth and Ozaukee County and Port Washington as well too. So we're using this regional model, this regional group and association to again further resources and information sharing to these Infos project sites. NOAA leadership site visits. We've had an opportunity to bring senior leadership from NOAA to City Port Washington to talk about a multitude of projects, but we also highlighted the Infos work. And we talked about the leveraging of all these resources, and it's been a nice example for senior leadership at NOAA out of DC and Silver Springs to see what Wisconsin is doing and how Infos is actually having an impact on this community. Future opportunities, we're talking about so people can access remotely the Infos websites. They can access the Infos website through their app, their mobile devices to check on current conditions, forecast conditions. We're actually looking at the possibility of a kiosk at the site as well too that would have a tablet that people could tap into if you don't have a mobile device. Information would be there, and so we're looking at additional education and interpretation and access to the Infos website. So we're looking at installing a kiosk eventually in the city of Port Washington. The Port Exploring Lake Michigan Learning Lab, I'm working with them on their education curriculum. They want to tackle dangerous currents as an issue, among many other issues. And so they're developing curriculum right now. And so there's an opportunity to take what we've been doing and take the data that's been coming in through the Infos project and make that available to talk about the science of it and the public safety issues of that with school children in the Port Washington School District. We're prepared and looking at, we've talked to City of Port Washington about actually reaching the schools through the district to reach into the classes. We've had information that's been broadcast through e-newsletters. And we've been talking about a public information meeting at some point in 2017. And all in all, this kind of comes back to resiliency, is how do we make these cities resilient? How do we make these beach site locations resilient as you know, an asset to coastal communities? So dangerous currents and hazards are part of that as well too. And so we're actually looking at potentially developing looking through the coastal resiliency program at NOAA of additional resources to address that issue. These are the websites that I know that I think Chin had displayed for you if you were interested. And thank you. And these are Jesse, Jean's, mine, and Chin's email addresses if you'd like to follow up with those with additional questions.
Any questions? Go ahead.
I admit I have no idea how technology will work to this, but is there any way that you can get your camera phone to integrate that technology so you can take a picture or a video of each that you're out in the lifeguard and then you tell time if there is a recurrence?
So the question was whether you can use your phone camera to see a rip current using technology?
Yes, I believe it. Engineering is the easiest. Usually you put it out, the more the people are coming. You just make it more work.
All right. You want to wrap it up, Shannon?
All right, well, thank you, Donnelly. We're all doing pretty well so far. There's a little bit of a snack left if you want some. And we're adjourned for so far today, but remember the posters are upstairs. They're going to be upstairs for a little bit longer. They're going to go down tomorrow, like 10 o'clock-ish or whatever. So if you have a poster and it's upstairs, you're going to be responsible for getting it. The easels will be taken away tomorrow morning. We're going to be back here tomorrow at 8:30. So come back for more. I don't think we're going to talk about QPCR a lot more, but we might. So there's some restoration sessions tomorrow and remediation. So thanks again. Have a good evening. We'll see you back here tomorrow at 8:30.